sì, prova, ehi. Adria, collega, guardo cavo per favore. Prova, sì, prova, sì, prova, ehi, sa. Adria, stando davanti su cavo e vediamo che te lo cavo. Va, prova, si prova, ehi. L'inglese è questo qua, è quello che abbiamo adoperato ieri. Prova microfonica, sì, ok? Prova, vabbè, lascialo così allora. Prova, sì, prova, perché mo è come dire, poi gli diamo l'input dal mixer, ok? Prova, ok. Non siamo noi. Prova, Andrea, poi magari sul volume... Dove ci arriva l'audio? Prova, ehi, sì, prova. Domenico, ciao Domenico, tutto bene? Dai l'input, Andrea, dai l'input sul... Sì, dai, aumenta il volume da lì, non basta che lo dici che aumenta. No, che poi rimbomba, Andrea, lascia così, sì.
Ma perché non vediamo nessuno qua?
Ci devo autorizzare il comando.
Ah. Ci devi autorizzare, Antonia?
Ok, possiamo anche...
Io sto autorizzando tutti quelli che
Anche ci sarà Gershon, io ti ho messo un video sul desktop per vedere. Questo,
Siamo subito. Quindi voglio spiegare dicendo che
tanto lo sanno, siamo pure su Meet, sono presi quindi la prima è una signora, si chiama Gaia Guarino.
Quanto c'è puoi già entrare? Quanto vuole puoi già entrare?
Pronto? Mi senti?
Cominciamo a partire, dai,
Aspetta,
partiamo?
allora mi vedeva
la paura.
Dobbiamo presentare la Gaia Novarino, la presenti tu,
Sì.
che ne sai di più di me. Io presento Evan e tu puoi presentare Gershwin, che sei più familiare con me.
La sera, siamo questa casa. E qui,
Domenico?
no, Comunque, lei sul computer di approccio, va bene. Con lo fa bene installazione.
Non mi lascia... Non mi lascia partire qua.
Un attimo che partiamo pure con me, facciamo tutte e tre.
Ma questo dovrebbe essere già collegato?
Quella che dice collegata. Antonia?
Sto mettendo un sacco di persone.
Fammi attaccare qua.
Sì.
Penso che le credenziali.
Ma la pancia l'ha presa. Perché così aumentiamo le visualizzazioni.
Ok. Adesso... Ciao Gaia.
Ciao.
Ciao, grazie di essere qua con noi. Solo un secondo che finiamo di collegare.
Certamente.
Eccola lì, Gaia Novarina. Buon pomeriggio Gaia. Buon pomeriggio.
Buon pomeriggio. Non vedo nessuno. Ah, eccoci.
Lo so, non ci vedo.
Ah, eccoci.
Grazie per essere con noi.
Mi dispiace di non poter essere là in presenza, ma purtroppo veramente non sono riuscita.
Vabbè, la prossima volta.
Sì, assolutamente sì, molto volentieri.
Mancare. Bene.
Volete che intanto provo a fare lo sharing dello screen?
Sì, proviamo.
Quando vogliamo aspettiamo che arrivi qualcheduno. Chi c'era che doveva arrivare ancora?
Non lo vedete, vero?
No.
Quanti sono collegati lo sappiamo, Franco? Sappiamo quanti sono.
Ma adesso stanno entrando nella piattaforma.
Allora aspettiamo un momentino Gaia.
Sì, intanto devo riuscire a fare, perché ci sono problemi per lo sharing dello screen, non è così?
Noi non vediamo ancora nulla.
Sì, lo so.
Hai un Mac anche tu?
Sì.
Perché certe volte, questa mattina abbiamo avuto problemi con Carlo Sala e certe volte è un problema di autorizzazione nelle policy di privacy. Però se hai già fatto presentazioni...
Però non mi ha dato, adesso controllo.
Chiedile da dove sei, a Vienna? Dove?
Sì? Sei a Vienna o sei in Italia?
Sì, no, sono a Vienna.
Università di Vienna si chiama?
No, è l'Istituto di Science and Technology, è un istituto, diciamo...
Di ricerca?
Di ricerca, sì. Sto contemporaneamente cercando di fare... aspettate che mi ricollego in un secondo,
Sì.
Eccoci. Bisogna parlare in inglese qui, a cominciare.
Sì. Io, vabbè, tanto si è presentato da solo.
Sì, vabbè, insomma, giusto dire due parole.
Sì, sì.
Riuscite a vedere il mio screen adesso, vero?
Sì.
E se faccio così vedete la presentazione o no?
Così vediamo solo il tuo screen. Adesso rivediamo te.
Sì. Ups.
Dunque, se
Forse
sei...
ci vuole un pochino di tempo perché arrivi anche a noi, sai, a volte succede che se tu metti in modalità presentazione c'è un piccolo gap di tempo che anche gli altri
può
riescono a
essere.
vedere.
Vedete adesso?
No, non ancora, io almeno.
No.
Questo è interessante perché non mi è mai successo.
Hai provato a presentare il tuo schermo intero?
Sì, quello che ho fatto prima è quando mi ha fatto vedere ma non mi fa vedere la presentazione. E tra l'altro poi mi va proprio via l'opzione, adesso non c'è più l'opzione di fare sharing dello screen.
Piuttosto possiamo fare come prima e ci puoi mandare la presentazione.
Sì, però poi vi devo dire ogni volta di andare avanti.
Sì, quello sì, lo posso cambiare io.
E tra l'altro è anche piuttosto lunghetta, cioè nel senso grande. Veramente non mi è mai successa questa cosa. Fatemi provare un secondo ancora, un secondo,
Sì,
una
vai.
cosa.
For how long are you going to speak, Gaia?
Microfono spento.
Si sente. No, il microfono.
Yeah, in principle I was doing 1 hour because that's what was in the schedule, but I mean I can try to shorten it up. So in the schedule was saying from 3 to 4, but
Era un'ora inizialmente.
Non so se può essere utile, ma io stamattina potevo condividere solo la presentazione.
io invece è proprio quella che non posso condividere.
Ok, perché anche io ho un Mac, allora dicevo magari è mal comune. Niente.
Si può iniziare?
No, abbiamo ancora problemi di condivisione. Problemi di condivisione della presentazione.
Dai, intanto vi mando la presentazione perché altrimenti qui...
Aspetta che ti indico la mail.
Sì, ve lo devo mandare attraverso WeTransfer perché sennò non funzionerà mai.
Scusate, ma è impossibile che non... Ah, deve cliccare.
Però non funziona da lei. per qualche motivo non...
Che usa Mac?
Sì,
Dobbiamo fare come stamattina. Allora, andiamo questo, e ne facciamo andare così.
ok. Ti mandiamo una mail così ci arriva direttamente sullo schermo.
Grazie.
Poi ti apri questo, lo scarico. Ok, mi scrive intanto qua.
Fantastico.
Poi devi mettere.
Please. Well, there is a speaker, but if you can see and listen, try, or try it, otherwise you can move to a more convenient place for you.
I mean, I can put my computer on as well.
Yes, you want to put your computer about your talk.
Oh, yeah.
Il prossimo, c'è prima questa signora, e poi c'è questo signore.
Sto provando a entrare anche dall'altro
Ok?
Google Chrome.
Okay, very good.
Ok. Questo qua copiamo sopra.
Is it the first one?
Yeah, that's it.
Non vedete la presentazione ancora così, vero?
No,
Okay?
vedo sempre la tua telecamera.
Which floor is your room? First,
106.
106. Next, close to my. Okay, first floor.
What are you?
I'm 102. You have this Gaia Navarino first for 35-40 minutes. Quando possiamo iniziare?
Possiamo anche invertire volendo. Possiamo invertire?
Possiamo? Facciamo l'inverso?
No, mi dispiace, io ho un impegno.
Hai un impegno tu, scusami.
Alle 5 ho un impegno.
Ok.
C'è un problema.
Ce l'hai inviata?
You're ready.
Non ancora, sto cercando perché non lo devo fare attraverso momento che qui non sono troppo attraverso WeTransfer
Yeah.
e non riesco a ricevere
Ha un problema lei? Facciamo iniziare. We can start.
il codice.
Come? Non più. Ok, let's see if she can make it. She has a problem.
She cannot share the slides.
Well, could she email them to you and then you
Yes.
present them here?
Yes, I may.
Apparently she has a problem with the slides. Don't understand.
It could be the sharing program too.
Yes. Hai provato a mandarcela, Gaia?
Sto provando,
È
ma
grossa.
sto provando attraverso WeTransfer, ma non riesco, non mi riceve adesso, ovviamente non funziona niente, non ricevo il codice per mandarlo ancora.
Perché sarà grossa.
No, sì.
Perché non fa?
Adesso, aspettate.
Forse sono troppo pesanti? They're too heavy.
No.
Is it too big to send?
Ok, adesso fra pochi secondi vi dovrebbe arrivare.
Ok.
L'altra possibilità è che se volete io vi posso fare sharing di un Zoom link e lì funziona tutto quanto e tutti si possono ricollegare lì come volete.
No, non riusciamo, così dovremmo cambiare la piattaforma e non riusciamo.
Ce la dovreste avere ve l'ho mandata adesso.
She can send us a zip or a PDF.
Ha mandato, sì, l'ha mandata, dice.
Vi arriva attraverso WeTransfer perché è grande, altrimenti per email non arriva.
Ok,
È arrivata?
non ancora.
Guarda nello spam.
E questo?
Eccolo qua. Ok.
Ok. Here are the slides now.
Ok, ci sono quasi. Ok, we're almost there.
Even in
It's
Vietnam.
something that doesn't work, of course. This was
No.
not like this, but
Okay, I think it's working. I can change the slides for you.
yeah. Yes, I mean, this was, there are probably some problem because this obviously was not like this, but doesn't matter. We go on and try to be fast and see how this goes, okay? It's already late. Thank you for inviting me and sorry for those technical issues. It has been a while that I haven't had issues like this. Over the day, I mean today, this morning already, and later in the afternoon, you will hear a lot from genetics of autism spectrum disorders. And so what I thought to do today is a little bit to show you what we are doing in a way to sort of move from the genetics to the physiology and phenotypic outcomes of genetic mutations. So next. And so in general, I'm sure that you all know and you already got an understanding of the fact that autism spectrum disorders in general are very heterogeneous disorders from a genetic point of view. Next. And so what we are trying to do is really to try to understand how different forms of autism spectrum disorders, they may share either molecular mechanisms or phenotypic traits. Next. And so indeed, I mean, this is not primarily our work, but over the years we have found, and Carlo Sala talked about this, that for instance, genes linked to synaptic development and plasticity and function eventually are linked to autism spectrum disorders. Next, but there is also a number of other genes and linked to other molecular functions such as transcriptional regulation and proteostasis that over the last few years have been linked to this group of disorders. And so recently we became interested in trying to understand how those different classes of genes and or forms of autism spectrum disorders may in fact even be linked together with each other. And so what I'm going to talk about today, next one, is about how eventually a mutation in genes related to transcriptional regulation and chromatin modification or histone modification may be linked to the synaptic deficits that we know to be linked to autism spectrum disorders. And so for that, I named this talk then Genomic and Synaptic Plasticity in ASD. And in fact, as a neuroscientist, so I am a neuroscientist, a neurogeneticist, but a neuroscientist primarily, we often as neuroscientists think about the brain as the synapse as the primer place of the most important thing that happens, the most important organelle, if you want, that the brain has. But in fact, we know that a lot more is happening in the nucleus. And so what we're really trying to understand, next one, is what is the connection between the nucleus, what happens in the nucleus, and what happens at the synapse. And all of this, what I'm going to tell you today, started a few years back when we and others identified mutation in this gene, SETD5, in a number of patients with autistic traits, as well as intellectual disabilities, some speech delay, short stature, and strabismus. So in the meantime, this gene has been described also by others as one of the often mutated, de novo mutated genes in autism spectrum disorders. And what we knew is that this gene is relevant to modify histone structures. So the histones are those protein that are used to pack the chromatin, so the DNA in the nucleus, and they need to change conformation in response when to allow gene transcription or gene repression. So SETD5 is one of those genes that enable basically the modification of histones and either closing or opening the chromatin. Next one. And so what we discovered back then is that SETD5 is interacting with a number of different complexes at the chromatin to regulate the transcription of neuronal genes. Next one. And in fact, so what happens is that while in the wild type, this process of transcription of neuronal genes is very well regulated, in SETD5 mutants, there is an exaggerated transcription of genes related to neurons or brain development. Next one. And in fact, so it's in a way, the way that we are seeing this is a little bit like this. A swimmer here that just simply starts earlier to swim, and so he's still doing everything fine, but it's just the timing is anticipated and or is not correct, and so then mutant for SETD5 eventually start to transcribe neuronal genes at moments that those neuronal genes are not needed. Next one. As a result, for instance, in the mouse, what we see is that next one is that there is, and here there is a conversion problem, but what there is that there is an expression, exaggerated expression of genes that are related to the head development and the central system development, and there is a repression and so a downregulation of the expression of genes that are related to other parts of the body. So in fact, if we look at the mouse, next one, if we look at the mouse, what we see is that the mouse model with mutation in SETD5 has a much smaller body than in the wild type. So on the right side, there is the mutant mouse, and the left side, the wild type. And you can see that the body of this mouse is much smaller, while the brain size remains normal. So really, kind of indicating, it's a good representation also, even if it's a simplification, to the fact that there is a depletion of expression of those genes that are related to various tissues in the body, but the brain instead maintains its normal size. Next one. And in fact, one of another feature that those animals have is this a white belly button here. So you can see that the hairs in the belly, they are white. And as a neuroscientist, usually to tend to sort of disregard those phenotypes. But in fact, this is a very interesting developmental phenotype indicating that there is from very early on during development, a depletion of the melanoblasts that are the precursor of melanocytes during development. So there is a depletion of those cells, and we know that this is because, in fact, there is a sort of bias of cell fate toward what are more neuronal cells rather than other tissues of the body. So at the behavioral level, what we found, next one, we found that, and you can move farther, so do another one. So what we found is that mutant for SETD5, they do have a problem in learning, but in fact what they show is an excessive, exaggerated sort of learning, so an enhanced learning, as measured here by this behavioral test that is called context sphere conditioning, where we are conditioning an animal to remember one context as an adverse context, and we measure the memory of the animal by measuring the percentage of freezing of this animal. And what you can see here is that the SETD5 mutants, they show this exaggerated freezing. However, so if you go next one, however, what we see is that when we apply a protocol of extinction that is in practical, let's say, to simplify, is a protocol to induce the forgetting, it's not completely correct, but just to make it simple, the forgetting of this adverse experience, what we found is that the SETD5 animals, they have problem in forgetting this, while the wild-type animals can really forget that this adverse context is not anymore dangerous for them. And so we read this analysis as a sort of, those animals are learning much better, but in fact they're becoming inflexible. Next one. And we can also see this at the electrophysiological level, but I'm not going to go through this, explain this, try to cut on the time. So next one. So what I say at the beginning is that what we are trying to do is to link what happens in the nucleus with what happens in the synapse as a way to link also why mutations in genes related to chromatin modification and as well genes related to synaptic functions, they both lead to out-in-spectrum disorders. So in order to understand this in the context of SETD5, what we did is an experiment to try to stimulate the function of the synapse and read out what happened at the level of the, let's say, of the nucleus. In fact, it's at the transcriptional level. Next one. So practically what we did is put an animal in a context where they experienced this adverse, where they had this adverse experience, and then 1 hour or 3 hours after this experience, we obtained their hippocampus and perform RNA sequencing to look at the expression of number of genes. And what we find is, in fact, that even in the wild type, this adverse experience leads to the expression of a number of different genes, and that the expression changes across time. So what we did is clustering those genes based on their trajectory over time, and so there will be some genes that are expressed immediately after the experience of this, this adverse experience, and then they will be downregulated afterwards. The other genes that instead they are increasing their amount steadily across time for a few hours after the experience, etc., etc. Next one. And so what we obtain is a number of different clusters. So each of these clusters contains a number of genes that are expressed across time, so after this adverse experience. And what you can see here is in gray is always the wild type, in yellow is the mutant. And what you can see is that, in fact, while there are a lot of genes that behave in a similar way, like those one in the, you cannot see my mouse, but the ones that they are at the bottom, for instance, the last two clusters, where yellow and gray lines, they are overlapping, indicating that those genes follow the same trajectory across time. After the behavioral test, there are a number of other clusters where a number of other genes where the two genotypes, they behave essentially differently. And what I would like to highlight, next one, are especially two clusters here, where you see that the yellow lines sort of keep going up, despite the gray line after 3 hours after the behavioral performance or behavioral test or training, it goes down. So they have this sort of exaggerated expression across time, so they are not able to shut down the expression of those genes in the mutant again. And those genes, next one, those genes in fact are very interesting because they are all related to synaptic functions, and they are depicted here either in orange or in red. And those genes are, as I say, related to synaptic functions and often also eventually mutated in autism spectrum disorders. So practically what this tells us is that SETD5, when mutated, leads to an abnormal expression of a number of synaptic genes, and that those synaptic genes are eventually also linked to autism spectrum disorders. Next one. So in a way, what I have shown till now is that a few years ago we identified mutation in a gene called SETD5 linking autism spectrum disorders, intellectual disability, and other features. That this gene is linked somehow to the modification, or actually to the function of the histones to pack and unpack the chromatin to make it available or less available for gene transcription, and that when SETD5 is mutated, the transcription of genes related to neuronal functions and synaptic functions is somehow abnormal. Next one. And so what we wanted to do now, it was really to understand whether this is a unique, is a feature unique to SETD5, or in fact is a feature also associated with mutations in other chromatin remodelers. And so for this, we really decided to go to be a little bit more systematics, and so next one. And so what we did is to look. What we wanted to do is on one side see whether mutation in those chromatin remodelers are affecting just this form of Hebbian plasticity, so the one that is associated to learning. Sorry, there is a typo, but that is associated with learning. So that so-called Hebbian plasticity, that is the one that you had just showed for SETD5. Or next one, they're also associated with another form of plasticity that is called homeostatic plasticity, that is a way to sort of keep the synapses in a sort of dynamic range. So in a way you can see this process like when you are storing a lot of information in a computer, at one point eventually you get to a place where you cannot store anymore because there is not sufficient memory anymore. And so a way to overcome this is practically to cancel and to remove or to sort of scale down those synapses or your computer to make space available to eventually put more information. Next one. And so in order to you can go next one. So in order to be systematic, what we decided to do is to generate mouse models for 4 different chromatin associated genes. So SETD5 has shown LKDM6B, KMT5B, and so those are all what they are considered sort of high risk ASD genes. So a SCORE1 Safari database, and so we generated for those always constitutive heterozygous mutants. The patients are all heterozygous, and we also in parallel did those inducible reversible lines that we will see later why we are using and for what we are using. And so using those animals, next one, what we did is look at, study the avian plasticity as well as homeostatic plasticity at the behavioral and the electrophysiology level, as well as the genomic level. So looking at how gene expression is regulated by using RNA sequencing and data sequencing. However, since one of the main complaints for those that are using animals to model autism spectrum disorders, and I mean in general we are obviously very well aware that we are not making autistic mice. This is not what we are doing. We are really trying to look at molecular mechanisms that may be conserved. Nevertheless, to really understand whether those are mechanisms that can be conserved in human, what we decided to do also, next one, is to generate for the same models also human cerebral organoids carry mutation in the same genes, so also heterozygous mutations, and here also use electrophysiology to study synaptic plasticity, such as homeostatic plasticity, and genomic plasticity using RNA sequencing and data sequencing. So what I'm going to do now is guide you through a few of the results that we obtained, and we are very excited actually, because we think that this can be really a way to kind of identify commonalities between different genes that are mutated in autism, and also identify eventually potential path for treatment. So next one. So what we did is always starting, or what we do is always starting from the behavioral analysis to try to understand whether those animals that have behavioral phenotypes that can be associated with the synaptic plasticity defects. So also here again, what we use is context-free conditioning. Once again, so this is a test to understand how those animals are learning. And so you are placing an animal in a context where you are delivering an adverse experience, and then 24 hours later, you basically test the animal to see whether they are remembering and recalling the fact that this context was not a positive context. Next one. And in fact, what we found, next one, what we found is that here, for instance, I showed the results for H1L and KDM6P, that in both cases, the animals, they are freezing less, so significantly less, indicating that those animals remember less about that this context was adverse. So next one, and this is one. This is also visible by performing electrophysiological analysis, where we see that the electrophysiological or physiological correlate of learning, such a long-term potentiation, is affected and is abnormal in those animals. So it's much reduced in those animals. Next one. And so what we did then was using RNA sequencing as well as morphological analysis to see whether those animals at the baseline already, so before their learning experience, may have synaptic deficits. And what we found is that, in fact, there was not a very, so there were no morphological issues at the level of the synapses. So the synapses, they look pretty normal in those animals, and also there are no major differences at the level of gene expression at steady state, so before the animal has this sort of bad experience. Next one. However, so because we want to understand this communication between the synaptic function and the nucleus, we repeated the experiment after looking at gene expression after we trained the animal for remembering this adverse experience. Next one. And here, what we found, what we basically repeated is the same thing that I showed to you before in SETD5. So we placed an animal in a cage where we deliver an adverse experience, and then 1 hour and 3 hours after, we obtained the hippocampi and did RNA extraction and did RNA sequencing. And what we found is that, once again, similar to what we found when we were doing the project for SETD5, is that in the wild type, there are a number of genes that are increasing their expression. So below you can see those red and blue bars. I'm sorry that I cannot point them out where I want to really indicate, but so what we see is that 1 hour after the training, there is an increase here indicated as a red of about the expression of about 400 genes, so about 397. And 3 hours later, there is, in parallel to an upregulation of genes here in red, those 656, that are increasing in level, there is also a downregulation of a number of other genes. So about 1,000 genes are repressed during this learning process. And this is very interesting to see this very strong gene repression, and what we interpret, so the way we interpreting this, and others have seen similar things as well, is that in a way that there is a first wave of gene expression that is really associated also with this process of learning, but then there is a wave of repression that is sort of needed to put a break in the process to really tune the synaptic plasticity events to that time frame where the animal is experiencing something. So this is what happens in the wild type. Next one. And in the mutants, what we found is that the response was much milder. So the number of genes that are either up or down regulated is much more limited. So 1 hour after the event is maximum 100 genes, more or less, that have a different expression, and 3 hours after also there is really just a fraction of the genes that we see that are regulated in the wild type that are also regulated in the mutant. Next one. So when we look at the function of those genes that are deregulated, what we found is that, or that are regulated within the process of synaptic, of Hebbian plasticity, what we found is that in the wild type, so this is what we are looking now, just wild type animals, on the left side, we see that, that we have an upregulation in red of genes related to gene transcription and histomodifications, as well as, so this is 1 hour after the training of the animal, while 3 hours after the training of the animal, we have the downregulation of those same genes. So in fact, a lot of genes that are related to gene transcription have a biphasic trajectory, whereby they are increasing in quantity after the training of the animal, and they are decreasing again in quantity 3 hours after the training. However, in parallel to these genes related to transcription, 3 hours after the training of the animal, we also see here in the right side an upregulation of a number of the transcription of a number of synaptic genes. So in the mutant, next one, in the mutant, what we observe is that the initial wave, so at 1 hour, of those red dots that are related to the genes that are related to gene transcription and histone regulation are completely absent. So there is not this enrichment for genes related to gene transcription. Next one. And also what we fail to see is a downregulation or a regulation of genes related to synaptic functions. So in a way, again, what we find is that mutations linked to chromatin remodeling are leading to a normal gene expression during the process of Hebbian plasticity, that is this form of plasticity that is related to learning. Next one. And so, as I mentioned before, what we wanted to understand is that this is only related to the learning process or also to other form of plasticity, so more general plasticity as homeostatic plasticity. And in order to understand this, we use another paradigm in mouse where we are basically closing one eye of the animal, and in this way eliminating all the input that go to this eye, to the animal, to the visual cortex and the other hemisphere. And so this is a way to sort of eliminate all the activity. And when the neuron realize that there is much less input and much less activity, has to bring the synapse back at the level and a range where it's functional. So what we know as upscaling. So next one. And so indeed, this is what we see in wild type animals. So you can see here in gray that when we are closing an animal, that here the eye of an animal that here is indicating with TTX, we see this in the histogram. You can see that there is this increase of amplitude of excitatory postsynaptic currents that are related to practically bringing the synapse to a higher level of activity. However, what we found is that next one, that this phenomenon, so it doesn't matter to go into the detail, but this phenomenon is also completely absent in H1L and KDM6B mutants. So you can really look just here at the histogram in red or in purple that those animals, when treated with DTX, they have a problem to making this upscaling of the synapse properly. So next one. And so in order to understand, and this is what I mentioned before, to understand whether this is in general is a process that is also conserved maybe in human neurons, what we did is, as I mentioned before, generating organoids. So those are stem cell-derived model system, so where you can differentiate, and I believe then Gashvin later, I looked over his talk, is also going to talk about this a little bit, how to use stem cells to model those disorders. And so that's what we did here. We obtained cells with mutation in the 4 genes that we were studying, and from them generated the cerebral organoids that are those 3-dimensional structures of neurons, of human neurons that you can study in various ways, including electrophysiology. So in order to understand whether this problem plasticity is also conserved in fact in human, in the presence of mutations in those genes, we treated those organoids practically in the same way as we treated the animals, applying TTX, and then look at this phenomenon of synaptic plasticity. Next one. And indeed what we found is that, again, you can just look at the histogram here on the left side, that TTX treated organoids that are wild type, you see that this increase in the amplitude of the electric activity, if you want, just interpret this in this way. So because they are trying to compensate the fact that we are trying to block their activity. So really indicating that also in the organoids we are able to induce this phenomenon of homeostatic plasticity. However, next one. However, this doesn't happen at all in all the mutants that we have studied. So here you see KM55B, H1L, KDM6B, and SET5 that all lack this property or try to compensate the TTX treatments. So they really show that they're unable to do this synaptic upscaling in response to a reduction of the activity. So this is quite nice because it really tells us that this is not just something that happens in mouse, but eventually happens also at least in human cells. This gives us a tool eventually to screen for potential drugs, and I will show this in a minute. So next one. So in order to understand a little bit better again what happens at the molecular level, we also did again RNA sequencing and ATA sequencing. Maybe I'm gonna, because of the time, because we started so late, and just trying to shorten up, so maybe we skip a couple slides. Next one. Next one. So I will just go here and tell you that what we found at the molecular level is that, again, H1L is unable to induce the expression of a number of different, so if you look at the RNA sequencing graph on the left, is unable to induce the expression of a number of genes during this other form of synaptic plasticity, while in this case, in the mutant of KDM6B mutant, we have a sort of an exaggerated program that takes over. And so this to say that 2 things. So first of all is that despite we see some convergences on the fact that at the phenotypic level, neither of the mutants that we are studying are able to induce the forms of synaptic plasticity that we're looking at, such as avian plasticity and homeostatic plasticity. At the molecular levels, they are not always behaving in the same way. So whereby, for instance, in the hippocampus, so one region of the brain, mutations in those genes that give to during synaptic plasticity, they lead to a sort of shrink and attenuated sort of nuclear response, if you want, while in another form of plasticity, such as the form of homeostatic plasticity, what we observe is that the H1L also again showed this attenuation of response, while in KDM6B, we found practically an exaggerated response that when we look at and analyze it well, we found that it's simply a response that is too noisy. So in both cases, what happens is that at the phenotypic level, they're converging, but at the molecular level, the mechanism seems to be slightly different. Next one. The next. So practically what I have shown here is that we can use mouse model as well as human cerebral organoids to try to understand how mutation in chromatin remodelers are affecting the expression of another class of genes that is linked to autism spectrum disorders, that is this class of synaptic genes that I believe you heard earlier this morning by Carlo. Now, what we had the interest, and I will take still a couple of minutes, no more, one of the reasons why we are doing this, and one of the reasons that, in fact, we used animals, and we used animals that were already adults, it was to understand whether mutation in chromatin remodelers are linked to just to developmental defects, or are also linked to processes that happen throughout life. So the process of synaptic plasticity is something that, so we always say, well, synaptic development and synaptic plasticity, so, but it is not limited to a specific time window. So it's not just happening early during development, but synaptic plasticity is happening throughout life. So whenever you are, for instance, learning something, but also whenever you are resetting basically the synapses. And so, in a way to see that those animals have phenotypes at the level of synaptic plasticity gave also us some hope that maybe those defects that can also be treated and ameliorated later during life. And so in order to understand this, what we did is creating those, what here are indicating those stop animals, so H1L plus stop or KDM6B plus stop, that practically are mutant animals that you can revert to wild-type animals at will, so whenever you want. And so the last data I'm going to show to you is the data on one of those animals, next one, where in fact what we are doing is when the animal is about 4 weeks old, we are injecting tamoxifen that is enabling the transformation of this animal from a knockout or a stop animal to a wild-type animal. And after that, the next one, we are looking at the expression of the genes, so like in this case KDM6B, which in fact increases its expression to go, so this is the most right box plot in the graph, so that goes basically to an expression that is closer to the wild type. And with those animals, what we did is repeating those homeostatic plasticity experiments that we did before with the mutant animals, next one. And what we found is that, in fact, injecting, so re-establishing the expression of the gene is also enabling the re-establishment of synaptic plasticity. And this is what you see in the right graph, where the turquoise plots that the amplitude side indicates the mutant animals, and the one with filled squares is the mutant where we rescue the level of KDM6B. And in fact, this rescue works very well, and if you would overlap this line with the one on the left side, that is the wild type, you see that they are completely overlapping, indicating that reestablishing the expression of KDM6B later in life is sufficient to overcome those synaptic plasticity defects that we observe in those animals. Next one. And because we have also the human cerebral organoids, what we did also is starting doing, using those parameters that we have studied, we started using the organoids to do drug screens to understand whether we would be able to identify potential drugs that could be used in eventually for rescuing those synaptic plasticity defects that we have observed. And the next one, and so what we did is based on the molecular functions of the genes that we were studying and the RNA sequencing data that we have obtained using both cerebral organoids and mice, we have selected a number of drugs that we predicted to be able to eventually rescue the phenotypes in a gene-specific manner. And here what I'm going to show to you is the functional, how this ANC1999 works in rescuing defects in the gene-specific manner. Next one. So what we did is using organoids and in using in those organoids synaptic plasticity as we have done before. So here you can see on the left side of the middle graph, the bar graph on the left, the black one is the wild type, and you can see again what we have shown before, that by treating those organoids with TTX, we are able to use the synaptic plasticity, and also when those organoids are treated with this drug ANC1999. And on the right side, you see the organoids that are just treated with vehicles, so now with the drug, where we are unable to induce this synaptic plasticity. Next one. And you can see that while the treatment with ANC is not able to do anything to rescue the phenotype in H1L mutant, is actually sufficient to rescue the phenotype in KDM6B. So indicating that despite those genes are, mutation in those genes are converging at the phenotypical level, and despite they have also some overlap at the molecular level, we still can see that the treatments that we identified eventually have gene-specific effects, because in fact they are not completely overlapping. So still we have really a lot to do to understand how mutations in different genes, they are converging. And so what are the points of convergence as well the points of divergence. Next one. And so what I've shown to you a little bit is how we go from the realm of identification of genetic mutations that I believe you are going to hear a lot today afterwards, and specifically how mutations then in genes that are related to chromatin modifications, they are linked to eventually to synaptic functions and to the mutations of another class of genes that are those synaptic genes. In this specific case, what I think what we found is that mutations in the chromatin remodelers that we are looked at, indeed they are all leading to a change in the synaptic plasticity processes, so on the synaptic functions of both mouse and human neurons, and also we found that the modality and the mechanism by which those genes are regulating those functions can be slightly different, although there is also a lot of overlap, and that eventually those can be a useful readout to screen for potential drugs to use for treatment. So the final slide, next one, is for the acknowledgement. So this, the one that I've shown to you is work mostly from Bernadette Basilico in the lab, that she was electrophysiologist and behavioralist, as well as with the help of other people as Christoph Doctor and Jessica Kirchner that helped a lot with the molecular analysis. And I'm sorry that I was trying to shrink everything a little bit because we started so late, but I still hope that maybe you could capture something from a little bit. So thank you for your attention.
Thank you very much, Gaia. It's really... Yes, I think we received some questions while you were speaking. I just need to... Ok.
Chiedi se ci sono domande.
No, this is a question from yesterday.
Messages.
Do you see any question in the chat? No.
I don't. I mean, I see one, but I don't think it's related to me.
No, it's something from yesterday, I guess.
Yeah.
Yes, sure.
La puoi leggere?
No, it's not a question for her.
Come?
Non è una domanda per lei.
Non ce n'era una? Prima.
No.
Non ci sono.
Can we ask
No
questions?
question.
Yes, we can ask questions.
Hi, this is Evan Eichler. Nice to see you again.
Hi, Evan.
That was a great talk. Just a question for you, and maybe you mentioned it and I missed it. Did you look at any sex-specific effects? So specifically, some of those genes that you have there, there's very weak signals. I wouldn't say they're significant, but biases in terms of gender, or sex, I should say. Did you match, when you did these mouse studies, by sex, and did you compare males versus females?
Thank you for the question. Yeah, absolutely. So I didn't mention, but we always, and I recommend that everyone should do this, always use both males and females, and we always analyze them together and separately as a way to really understand whether there are sex differences. So on the phenotype that they've shown you, so I merged the two sexes because we did not observe any difference. However, and I'd be more than happy to discuss that with you at one point. What we did, and this was a bit too much to show this, is we now have done a single cell sequencing analysis of about 12 different genotypes in mice across development. And we think that there we see a quite remarkable, and we are still analyzing this, but in some genes we see a quite remarkable sex effect, while in others this is completely absent. And some of those genes that they've shown also have this sex effect. So I would be super interested to see and look back at those genes that you see a signal or this weak signal that you mentioned to try to understand whether this match. I think that in a way the behavior is too, is not that precise, because there is a certain variability, even the wild type, to be able to detect very, very small differences when you are, looking at 20 animals. So probably you will be able to capture that if you would do cohorts like 100 animals, which we are obviously not doing. But this is certainly, we see something that is quite surprising in mouse to see these sex differences.
So I'm curious, both ASH1L and KDM6B show slight effects. And are those the two that were showing differences?
Look, I don't remember, because as I say, we are analyzing 12 genes at a time, and so this is tons of we did single cell sequencing, different brain regions, so we have about 400 samples that we are processing with the single cell RNA sequencing. So, but I look it up, and with my people in the next day, I just send you an email because I would be also interested to have feedback on that.
Yeah, thanks.
Okay, thank you. You have also a couple of questions from Alda Maria.
I can see. Yeah, so how do you relate melanocyte with brain synapses? So this is actually a very interesting question. So as I mentioned, when we started seeing, in fact, this was one of our animal caretaker that came up with this white belly spot. And at the beginning, we thought that has nothing to do with, neuroscience, so why we should look at. And as usual, this could have been more wrong, because we always, as researchers, we have to keep eye on everything. And I believe not just as researchers, probably also as a parent, we have always to try to observe everything. And so in a way, yes, this is in a way related. And it's related, of course, in a patient, you will not see necessarily white belly spot, but in mouse what happened is that those melanoblasts are unequally distributed across the body. And so the first place where you see this anomaly is on the belly. But melanoblasts derive, in fact, from the neural crest. And the neural crest is essential to make up a number of different tissues. So indeed, and in a way it is linked because in SETD5 we see a depletion of cells related to neural crests such as eye, teeth, and some other tissues. And we have this exaggerated expression of the brain. So no, it's not directly related to synapses, but still it could be that some of those dysmorphism that we observe in patients and they are also related to, in a way, gene expressions issues across development. So related to the genetic background of the mice, yes, this is obviously also a good question. We are trying to do also to analyze this a bit, but we know that the genetic background of the mouse influenced the results. But of course, because we're also working with organoids in parallel and we see similar things, I think that the phenotypes we're working with, they're quite robust to really say that are not influenced too much by the genetic background. But definitely this is something to take care.
Okay, thank you very much.
You have to show Evan how to maneuver the slides.
Okay, thank you very much, Gaia. And
thank you so much, and sorry for the technical issues.
We are indebted with you for sharing part of your Saturday with us.
No, that was a pleasure. Thank you so much.
Thank you, Gaia. Thank you very much.
Thank you. Bye.
Our next speaker is Evan Eichler. We are very, very grateful to Evan because he just arrived a couple of hours ago after a long trip through Europe, essentially, coming from Seattle 2 days ago. But I guess he had only 4 hours of sleep in the last 2 days. So Evan, we really do appreciate your effort to join us, and please go ahead.
Yeah, thanks, Giovanni. It's great to be here. As always, visiting Italy for me feels like a second home sometimes. I've been here so many times. So what I thought I would do today is give you my perspective based on data regarding essentially the genetic basis of autism. And I'm going to emphasize, as you will see, a particular class of variant throughout this talk to advance. Alfredo, do I just hit the... It's not advancing.
Do they see?
Oh, just a return. Or you can use these arrows. Wait, there's something wrong here. It went to the last slide. Yes. So it's not the enter button? Oh, okay. All right, so you probably don't need any... Wait. Okay.
Clicca sulla cosa. Ok.
Okay. Okay, so I'll start again. I'm going to talk today about the genetics of autism with a specific focus on rare variation. Make sure you get this right. There we go. I'm sure this has been discussed a lot at this meeting again, but a few points about autism. All the data for the last 50, 40 years has suggested extremely strong genetic heritability. That is to say, autism does run in families, and there's a genetic predisposition that exists in many families for this. The frequency of the disorder has gone up, just in part because of redefinition of autism. And I guess the other important point is that autism almost never sits alone with respect to disorders. It often is comorbid with others, such as intellectual disability, developmental delay, epilepsy, and a whole host of other often developmentally related aspects. So whether you think about autism or you think about any type of disease, I mean, from a perspective of a geneticist, there are at least two important things to consider. The frequency of a variant, so is it rare, is it common, which is what I'm showing here on the x-axis, and the effect size. So how likely is it that a person that carries the variant will actually develop the disorder? And so the field of genetics often thinks in terms of common variation that collectively accumulate within a person, leading to essentially disorders. What I'm going to be focusing on is on the bottom right portion. Variants that are extremely rare are essentially private or even de novo within families, with potential to have an effect size that is very, very large. So everything going from the bottom up to the top for the very rare. So I'm going to skip about 10 years of research, and just to summarize a few slides on essentially why we know rare variation, in particular de novo variation, is so important. So early on in the beginning of 2000s into the middle of 2000s, we started to screen families with autism with microarrays. That was the standard genomic technology that people used. And we found, in many groups, not just ours, that essentially children with autism had somewhere between 2 to 3-fold more large copy number variants than essentially normal children in the population. In many cases, these events were de novo. That is to say, the parents did not have these large deletions or duplications, but they arose in the presumed sperm or egg of the parent. And so the child was born and had large pieces of DNA, often dozens of genes, either deleted or duplicated. So the idea here was quite simple, that when you actually, specifically if you lost a particular segment of the genome, you would be predisposed to developing autism because you've lost some critically important gene in that region. When exome sequencing became available, and that was around 2008, 2009, we began to sequence families with a very specific structure. I'm showing that here in the bottom right-hand corner. These are quads. These are individuals, the parents are unaffected. The child, there's one child that's affected in that family and an unaffected sibling. So we refer to them in the field as quads. I think you may call them tetrads. Some people refer to them as that. But these were a very specific set of families where there's no necessarily family history. And when we started sequencing these families with exome sequencing and focusing just on de novo mutations, so these are mutations that occur in the child but not in the parents, we observed something that was significant within the first few hundred families. And that's shown here to the left. The number of de novo events that were gene killing, i.e. loss of function mutations, were about twice as common in children with autism compared to their unaffected sibling, who was serving as a genetic control for 50% of the DNA. This was a really important find. It suggested that de novo mutations that lead to the loss of the function of a gene, haploinsufficiency, are contributing significantly to the disorder. The bad news back in 2015, and we started these studies, and there were like a half a dozen groups that were doing similar ones, is that we almost never saw the same gene hit twice. So that told us something else. It told us that essentially in different families, different genes when mutated, result in autism. So now I'm going to actually fast forward to work over the last 5-6 years, where we've got to the point of now routinely identifying the genes associated with autism. And I decided for this talk today, I'm going to talk about 3 things. I'm going to talk about de novo mutations because they're so bloody important in terms of autism, and how difficult it's going to be to discover the last remaining couple hundred genes. Second, I'm going to focus on inherited mutations, but of a very specific type, ones that are private to your family. So we refer to private mutations as ones that we've seen once and only once in families of thousands of individuals, and I'll show you why that's important later on. And the last, which I believe is the future, I'm going to show you the beginning of actually comprehensively assessing variation and methylation in children with autism, and some hints, and I'm going to stress these are hints, clues of where the remaining genetic basis or etiology of the disease is. At least some clues that are coming up. So I've already introduced to you the quad. Yeah? Oh, sorry. So I've already introduced to you the quad design and the trios. And so I'm going to talk about a study where we've combined data from children with developmental delay. So this is the DDD study from the UK. Matt Hurls, in particular, led this, with a study that was driven in the US largely by the Simons Foundation to sequence 15,840 families. So I'm talking about 47,000 families now with children with developmental delay and autism, where we have a primary diagnosis as either DD or we have autism. So what my former postdoc, now a faculty, did is he took all of these different families. In some cases we generated the sequence in the case of autism, or wherever possible we used the data that was published, or we recalled the data, so we would use the same computational pipelines to identify the de novo events. So I'm not going to bore you with the computational pipelines that we're using, but in essence what we're looking for is an excess of de novo mutations than you'd expect based on a model of mutation that we now think we understand pretty well on a gene-by-gene basis in these, across the human genome. And we're doing this to identify genes that show an excess of either missense mutations or loss-of-function mutations in the autism cohort. And we're going to break out essentially autism children from the ones that diagnosed with DD to see if there's a difference in terms of the types of genes that we're identifying. Okay, so that's it in a nutshell. So we published this the end of last year. This is our survey of the genes where we find an enrichment of de novo mutations. So just to orientate you on this plot, we call it a bubble plot, partly inspired by Satterstrom from, at that time I believe, from the lab of Matt State. The size of the circle represents the number of children from that set of 47,000 that had a de novo mutation, either a missense or a loss of function mutation in these. And so there's 2 colors here. The darker color represents essentially, or I should say the green represents essentially the DD cases. The purple, if you can see it, represents essentially the autism cases in this study. The significance is indicated along the y-axis. So the higher the bubble, the more confident that we are that de novo mutations in these genes are associated with autism. And the order here represents strictly the order in the genome that they appear by chromosome. And then these 2 dashed lines, this is a cutoff for false discovery at 5%. And this FWER is basically genome-wide significance. So every statistician on the planet would agree that any gene above this line is essentially an autism gene, at least if it carries these types of severe mutations. So looking at this, I think you can get an appreciation that not all genes are created equal. Some genes are contributing significantly more cases to autism and developmental delay than others. But you would be hard-pressed to find many genes in this right now, at least if you focused on the purple, that aren't seen also in the cases of DD. In other words, almost every gene that has reached significance for de novo mutations in autism is also a gene that has been described in the literature as a gene associated with developmental delay. Okay? And we all know, I'm sure we all know autistic children, and not all of them are severely handicapped. In fact, there's a very large range of children in terms of, that's why it's called a spectrum disorder, that are very high functioning. These de novo mutations, by and large, are not responsible for that category of child. Does that make sense? Because we're picking up genes that in other clinics would be called essentially children with developmental disability. And that's particularly the case in Europe. Many European labs really don't recognize autism the same way the US does. So here's that in another way. So this is just taking the de novo loss of function mutations in this box. This is taking the de novo missense mutations. And this is just plotting children with the 15,000 autism cases that we've analyzed versus the 31,000 that came from DDED and other studies. So if any gene was essentially an autism-specific gene, you should see essentially no cases of DD or ID, and you would have a value up here in terms of the number of cases. There is essentially no such gene that we've identified thus far based on our sequencing of 15,000. And I'm not sharing you the data, but we've sequenced with Spark another 15,000 on top of this 30,000, and we may have found our first one, but extremely rare. It needs to be validated. But by and large, we are not seeing in the children any evidence of a de novo mutation that is exclusive to a diagnosis of autism. Almost every gene that reaches significant is significant for studies of DD as well. And I just color-coded the genes and put some names up for those that genome-wide significant versus false discovery rate here for you guys to see. The flip side, however, is not true. Just to stress this, there are genes that are associated, in fact, there are many genes associated with developmental delay that we have never identified in a child with autism, because developmental delay is a broader spectrum of disorders, and particularly there's many different ways to get intellectual disability, or I should say many more ways than there are for autism. And that's shown here by these individual genes, which are significant and seen in many cases of DDID, but are zero in our survey of ASD. So no, or little evidence, I should say, with some caution, because obviously we need to sequence probably hundreds of thousands of families before we make that claim too strongly. And I'm saying this only from the perspective of de novo mutations, to be clear. All right, so a couple points, hopefully to tie in with maybe some of the other talks, including the one we just heard. So if you look at the functional network, so this is looking at protein-protein interaction of all the genes that we've identified. So this is that less stringent set of 615 that I showed a few slides ago. These are the pathways that we identify, the clusters at least, functional networks that are associated with autism. And you've heard about them already throughout this meeting. The biggest one, which is shown here, we call it cluster 1. These are all the genes that are associated essentially with chromatin remodeling and chromatin interaction. And the black ones, distinguished from the blue, are ones that are defined as essentially hubs. So genes where there's more connections than you would expect by chance. So in fact, these are all protein-protein interactions. That's what those red lines are indicating. And this is one very large, in fact, this is almost half of the genes that reach significance for autism and de novo mutation are in chromatin remodeling. This is this other category, we call it cluster 2 in our lab. These are the genes that were referred to as well in the last talk. These are the synaptic genes. So these are genes associated with synaptic plasticity, learning, processing. Many of those genes, many of them actually channel genes in their own right, are located in cluster 2. And then you'll notice, which I think is super interesting, there are other clusters that generally aren't seen as much in the literature, but for example, cluster 3. This is one that is associated, at least by ontology domains, with essentially insulin receptor, kinase function, phosphatase activity, and such. I should say that's cluster 4. Cluster 3 is insulin receptor. Cluster 4 is phosphatase. And then cluster 5 looks like it's GTPase binding activity, and so on. And then there's a whole bunch of other genes that don't fit beautifully into any pathway yet. Probably there's in fact incomplete information on this, and there's probably many more pathways and interconnections that need to be made over time. But I guess the important point here is the genes that are being hit in children with autism and developmental delay are highly non-random in terms of their function. This is another dimension on top of that, and this is work from our colleague Tom Nowakowski, who we've collaborated on this. He looked at data from single-cell RNA-seq data from 75,000 individual cells at this time when we did this analysis. And so what we did is we said, okay, let's consider our clusters, and let's see if there's any enrichment in specific cell types in the developing human brain. So this is shown here in this heat map are all the individual genes that are, or I should say transcripts that are actually reaching high levels in association with the clustering for autism genes. So when the color gets warmer, that means there's actually evidence of specific cell types showing an enrichment in these autism genes. And maybe not a surprise, genes associated with GABAergic neurons, these are inhibitory and excitatory, glutamatergic, they're enriched in many of the genes, including the synaptic genes that I showed. And then up here, you see some warmth in terms of the cell types that are being identified. These are in intermediate IPC as well as radioglial cells. Radioglial cells are progenitors for essentially the developing of neurons in the cortical layers of our brain. So on the right-hand side, I think is a neat analysis. It plots essentially the different clusters by their functional activity here on the x-axis with comparing in the rows the different cell types. And whenever you see a little star or a double star, it indicates that there's significant enrichment for that cell type versus that cluster. So it's a 2-by-2 comparison here. You see that inhibitory neurons as well as excitatory neurons are enriched in cluster 2, which makes perfect sense because that's your synaptic genes, and so they should be essentially expressed and interacting there. Cluster 1, which is the largest proportion of autism genes, is associated with many of these genes here, such as the radioglial early cells that are present and dividing cells and so on. These are ones that are associated with essentially the development and production of neurons. And then there's some really interesting observations, which actually neither Tom nor I have completely understood, but there's that C3, which is that insulin receptor pathway, associated with cell types, again, endothelial and mural. These are supporting cells in the developing brain that almost are never considered in terms of autism. And granted, it's a small number of patients of the grand total, but most of the emphasis in terms of developing or focusing on treatments and therapies are focusing on looking at excitatory, inhibitory neurons, or perhaps these as potential targets in terms of chromatin remodeling. So this is just to suggest that I think we need deeper sampling to understand essentially of both the genes as well as the potential functional cell types that may be necessary to target for essentially treatment down the road of this disorder. So the second thing, which may be relevant to some people in this room, is that a lot of the data that I just showed you was based on exome sequencing. And over the last 10 years, there have been cheaper ways to actually target subsets of genes that you're interested in, screen in larger cohorts. So this is technology called molecular inversion probe assays developed by Jay Shanduri, where you basically create oligos that have some spacing that's fairly tight, usually 100 or plus base pairs between them. And you can design these in such a way that you can generate thousands of these for your genes or your exons of interest. And then you can target 50 or 60 genes and you can resequence using these molecular inversion probes where essentially the ligase comes in and you actually close and you actually synthesize with the polymerase the intervening sequence and you feed it through a sequencer machine and you get sequence for your specific 50 genes for about $12 a sample, once you've gone through all the effort of building essentially the inversion probes and targeting. We don't use this anymore because we've actually used up all of our patient material, but we spent about 10 years building a cohort of 20,000 patients' DNA, getting consent to use that material. And it was all clinically gathered material that we essentially spent in terms of targeting, I think, 5 or 6 different probe designs. So we collected these samples from 18 different centers from around the world where there was diagnosis of either autism or developmental delay. And we actually used these 20,000 DNA samples to confirm and replicate genes that did not reach significance in the first part of this study. So genes that are at the borderline below the standard less significance levels. We could bring them forward and actually prove them early on. So it's a very powerful way, if you have a biobank of thousands of samples, and again, this is agnostic to the type of disease that you're interested in. If you have the samples to actually type, you can actually sequence the protein coding sequences, i.e. the exons, for about 50 genes for about $12 a sample, which in the world of locus heterogeneous disorders like autism is a big deal, because they're individually each is individually rare, but collectively as a class they account for a significant fraction of cases. So this is what the postdoc did after we did this first study looking at genes that had come up as potentially significant or borderline significance. He then pushed them through essentially a MIP design and actually did two things. He proved genes as being significant statistically, but then we also had clinical labs that had access to these 20,000 patients where they could recontact the 5 or 6 cases that we found for a new gene. Okay. And I'm not going to go into this. This is the replication that was done in those studies of basically going back and sampling another set of 20,000 cases, proving individual genes as being enriched in terms of missense or enriched in terms of loss of function mutation. And then from that, over the years, has come dozens of papers. Not all we're the senior author on, because there are a lot of clinical labs that want to run with a specific new form or new genetic cause of autism and actually pursue it as actually a research program in its own right. So this was actually wonderfully, collaborative, and it provided the opportunity pretty much for every clinical lab to start to write their own papers. This was almost before the days of gene matcher and other things which people often use now, which somewhat circumvents this. But if you have samples that are uncharacterized, there are ways to do this, and using molecular inversion probes to actually sequence and find and prove individual genes. All right, so when we were done with these set of studies, and we had kind of both theoretical and empirical estimates of what fraction of autism do these de novo mutations explain, the most optimistic numbers are about 20 to 25% of cases. The most pessimistic numbers are about 10 to 15%, and the truth is probably somewhere in the middle, okay? So that was a lot of work, a lot of money, and for all those families that we solved the case, it's fantastic, right? And having worked with those families, that has actually in its own right been some of the biggest reward, but that still leaves most of the genetic cause of autism as unexplained. So I'm not going to talk about polygenic risk score or common variance, but I'm going to go to a second class of mutation where the data are, I think in terms of the individual gene level, are less convincing, but at the aggregate level, I think is absolutely clear that there's a signal. And that is the ultra-rare inherited mutations. Okay, so what's an ultra-rare, or what I'll refer to as private mutation? So de novo, you all understand. Child has it, parents don't. It happened during meiosis or really early during development, such that the majority of cells carry it. A private inherited mutation is one that has occurred in the grandparental generation or the great-grandparental generation, somewhere in that time frame, such that when you search the existing databases or all other families, you find no evidence of that mutation being present. So we're looking for things, I think my colleague called them, she had a name for them. Oh gosh, it was a Latin name too, which means penultimate or something in Italian, not Italian, but Latin. Gosh, I wish I remembered it. Oh, diproximal. She called them diproximal mutations, which means that they occurred in a few generations right above. And so I had a postdoc come in and I said, we need to study this because there's evidence, at least I could see in the literature already, of these types of mutations. So let's do it systematically. Let's go back to the families that we have with autism, especially the squad design, because we have the affected and unaffected, and we can test this in a very simple way. So I've already mentioned what they are, and I'll just mention first the cohorts that we used. So in this time, instead of combining DD and autism, we focused exclusively on autism. We were part of an effort to sequence about 4,000 families that were quad designs for the most part, essentially with whole genome. And we also teamed up with SPARC, which is an effort funded by the Simons Foundation, which their goal is to sequence 50,000 families over the next few years. They're up to about half that number at this point. And so what we did is we took 27,000 individuals that came, or in other words, 6,500 families from the SPARC cohort, and we did 3,400 families from essentially the genome sequencing. And the test was very simple. We're going to look for loss of function mutations, severe missense mutations that are present in the parents, not seen in databases, so pre-existing databases like DBSNP that exist, and not seen in any other autism family, because we want to find events that we believe will be ultimately selected out of the population, but we want to find them very early during the kind of that family's history. And the test we're going to do is we're just going to compare the affected and unaffected in the family, once we've identified these variants in the parents, and see if there's a transmission disequilibrium. And transmission disequilibrium is quite simply, do more events go to the child with autism versus the unaffected sibling? So we're comparing transmission within the context of the family. It's a very kind of old human, I think it's not even a human genetic test, it's an old test geneticists have used for years. But we had here the quad design, which is absolutely critical for this. So this is the result. So this is the result from the actual individuals that are coming from the genomes. This is where the mother, father, the child, and the unaffected sibling were sequenced whole genome, 30x sequence coverage. And just to put this into context, what you're looking at here on this axis is the odds ratio. So that's, you can think of it as like the likelihood that essentially the transmission would favor, in this case, the way this is drawn, the proband versus the sibling. So if there's no difference, the odds ratio would be 1, right? Because it would be equally likely to go to the proband or the sibling. So I'm showing you here in red loss of function mutations that are private. And I'm showing you this kind of the 95% confidence interval around that. And then I'm showing you here on the x-axis the likelihood that the gene is actually going to be intolerant to mutation. So when there's a 1, that means the gene is highly intolerant to mutation. And 0 means our genome doesn't care whether essentially this is mutated or not. This, I think, is a super interesting result. So you'll see, first thing you should see is that the loss of function mutations in red are highly significant, right, for the child with autism, based on this sampling of 4,000 probands versus 2,000 siblings in these quad families. You'll see that missense and synonymous, there's almost no signal whatsoever. Synonymous is like our control. Synonymous changes shouldn't have an effect, and certainly they're basically as 1 as you can get. Missense, I don't know if you can see it, but there's a little bump here. And we think that little tiny bump there is significant. But I want to come back to this PLI test, because this PLI is very different than what you see for de novo mutations. For de novo mutations in children with autism, most of the genes, 90%, 95% of the genes are in a PLI above 0.9. For inherited private mutations, the PLI shows significance all the way down to like 0.25. So these are genes that by and large many geneticists wouldn't touch with a 10-foot pole because they're genes that tolerate mutation, but they're obviously being preferentially transmitted to our children with autism compared to our unaffected sibling. I think this is super important because I think it's telling us something fundamentally different about this type of gene. All right, so that was the observation in the CCDG. Those are the genomes. Then we just repeated to see if it replicated in SPARC. Those are the ones that were done by exome sequencing, quads once again, and that's the replication. Okay, so for geneticists, this is about as good as it gets, and so we were really happy with this individual result because it said that we now observe this twice independently in children diagnosed solely with autism. There's a private inherited component that's coming from extremely ultra-rare loss of functions that are transmitted to the actual child. The third observation from this is what happens if we take all the genes that we already identified with de novo mutations as enriched. Okay, so remember I had a list of 615 of them, and in fact, you can make a list of about 1,000 that people think are involved in autism for de novo mutation. So what I'm showing you here is again that constraint for the, and the odds ratio. But what I'm comparing here is comparing all genes, and this is then subtracting the genes that we've already implicated in terms of autism. So this delta that you see is the difference that happens when I take out all known genes with autism out of the equation. And I hope you can agree that the odds ratios barely bump. In fact, if you estimate it, the data suggests that 95% of the burden that's coming from private inherited mutations is in genes that we have not seen yet from de novo mutations. Maybe because they actually don't have a large enough effect to be seen in a de novo mutation sense. But this says it's a large category. So if there's 500 genes that contribute to de novo mutations with autism, there's some number of genes beyond that that are contributing to this inherited component. This is the most interesting result in my opinion, but, and I hope you guys can follow this, but this is actually calculating, essentially once again something that you've seen already, the actual numbers of probands versus siblings for just different PLI bins. So this is the intolerance to mutation. And even though you can see there's significance at every point in these categories, comparing the number of probands that carry a private loss of function mutation versus an unaffected sibling, you can see here that the significance gets a little bit higher when you get genes that are less tolerant to mutation. Okay, so it does matter to some degree, but you can see that it's significant all the way through. But these are the number of cases of the 4,000, that differential. So that in this case, there's about 1,000 individuals in this set that actually have these private inherited mutations. That's the difference, right? So in terms of contribution, it could be a significant fraction of cases. But if you ask a question, and the question is this, what if we do this again, but we ask that the child or the proband or the unaffected sibling have 2 or more of those private mutations? Okay, so that's going to ask that they have inherited from different genes a loss of function mutation that's really rare. They got it from great-grandparents on 2 or 3 sides, right, that have come into them. What happens is the significance gets higher. So you can see that obviously the fewer people of that type that have 2 or more loss of function mutations, but the differential between the probands and the unaffected sibling gets larger, and the result gets more significant. So what's going on? Well, what this tells us, or what we think it tells us, is that a mutation in this gene, loss of function, is not necessary and sufficient for autism unless it is compounded with something else. And so what's happening is that these mutations are arising in the family, they're transmitting, and because there are so many genes involved in chromatin signaling, genes involved in synaptic processing and plasticity and all these truncating or pruning aspects, the children are unfortunate in that they actually receive 2, they're compounding, and they're pushing that child over the threshold into autism. That's a model, but it would fully explain all the observations of the data. So, bad news. We don't have, or we didn't until like about 6 months ago or a year ago, we didn't have statistical significance for an individual gene. Even though the signal in the aggregate is there and it's reproduced, for any individual locus we can't prove, and certainly for the combinatorics of 2 or more, we can't prove any individual pair of genes. So out of my postdocs, desperate to know what the genes did, which I think is quite nice, she just asked a very simple, I thought was logical question. Let's look for all those events that we've seen that are transmitted only to probands, and let's exclude all known ASA genes, and let's try to stack the deck by keeping things at a high PLI. So let's look for lightning striking twice, so the same genes. How many times do we see them? So the genes in blue are genes with 2 hits, and genes with 3 hits are shown in red. And then this is the protein interaction network for those genes based on string DB. So this network, by the way, One could argue that maybe she should have done those cuts differently, but this network is significant for an excess of protein interactions when you do it this way, just based on genetics of autism in families. What I was most excited by was the fact that the actual pathways that are popping up are pathways that are the same pathways that we've seen before, or very closely related. So here's chromatin condensation and accessibility genes. This is an E3 ubiquitin protein ligase and proteasome production pathway. There's regulation of transcription, which would fit beautifully with a chromatin remodeling pathway. And then this one I scratched my head about because I knew nothing about these complexes, COP-B2, COP-A, and so on. But these are a protein complex that's important in terms of intracellular transport, actually in terms of production of moving molecules to vesicles on the surface of cells, which has obviously got to be important in terms of actually synaptic trafficking, right, and the development of those molecules as you go along. But these are all interesting candidates that I think would be worthy of pursuit. The trouble is that you have locus heterogeneity plus the potential for oligogenicity, which is basically this idea that you have to have more than one mutation or insult to push these kids into an autistic state. Last question on this one is we go back and we estimate the evolutionary age of the mutations that we see. Now remember, we only have 2 generations in these families. So we can't know if it came from great-grandfather X or great-grandmother Y. But what we can do is we can estimate the evolutionary age of these using a coalescent model. So there's a program developed out of Cambridge called RELATE. And what we're showing here is the average generations that are estimated for loss of function mutations that are going to essentially candidate genes in our probands. So the average age estimated was 2.5 generations in terms of the age of those loss of function mutations. These are other genes not implicated. Their average age is older, 3.7 generations. These are the ones in the siblings. They're almost twice the age that we're seeing in the siblings compared to the actual probands. So this is actually, again, consistent with our model, because the argument is that the mutations occur in generations de proximo, and they will transmit for a few generations until they cross paths with another mutation, leading to a severe phenotype and being removed from the population pool. So those genes are under selection because our proteins interact with other proteins and do not sit in isolation with one another. So this is a digenic, trigenic model of inheritance, not a simple one gene knockout results in autism, which is, I think, where 90% of the field is right now. So I told you that we didn't have individual genes with significance. The Spark study finally published their results independently of us, even though we helped them with some of their analyses. And I was just going to highlight that they observed both an inherited signal for extremely what they called ultra-rare mutations, as well as de novo mutations, which we've already discussed. But what they compared it with was the IQ of the patients that they had the data for, which they had collected for these, essentially for all the families that are coming in. And what I think is really interesting, and it's their observation, not ours, is they're seeing, and for these very rare mutations, they're seeing that the IQ is shifted up compared to essentially the kids that are carrying essentially the de novo mutations. In other words, those private inherited mutations are more likely to be associated with children with higher, I wouldn't say they're totally within normal range yet, because they should be centered around 100, but these are children that are essentially higher IQ than the children that we've discovered with de novo mutations. And then this is some genes, which I know very little about. NAV3, ITSN1, SCAF1, you don't see them typically on autism gene lists, but these are genes that have now reached significance in terms of actually excess of mutations present. Now remember, they're not going to be clean like present only in proband, not in sibling. They're going to be enriched and significantly enriched in proband compared to sibling. All right, so I'll conclude with this, which is where I think the field should go, but I don't think much of it has gone there yet. And it's to essentially go back to families where we haven't found a genetic cause and actually apply superior sequencing technology, long-read sequencing, to completely understand all the variation in those families, as well as methylation changes that are occurring in those families. And so I would just make the disclaimer, I have no stock in either of these companies, ONT and PacBio, so I'm a complete agnostic. I want both of them to win as much as possible so we can destroy Illumina. I'll go on the record as saying that, because Illumina has soaked so much money out of me and many other investigators over the last 20 years, they should be ashamed of themselves. So these companies have come up with new sequencing technologies that actually create sequence reads that are on orders of magnitude larger than essentially Illumina. And so the most interesting to me, in particular, is the Hi-Fi PacBio, because it's generating sequence reads that are routinely 20 kilobases in size as opposed to 200 base pairs in size that are as accurate or more accurate than Illumina technology. And so I won't go into all the things that this allows us to do, but some of you may have heard of the finishing of the human genome, the T to T, the pan-genome. All of it is being driven by the ability that we can now take any human genome, we can actually sequence both the paternal and maternal complement to near completion, going from one end of the chromosome to the other end, for really, to be honest, the first time. And I guess I would just want to tell you guys to stop and think about this for a minute, because it means that you're no longer discovering variation the same way as we've done before. We're actually building assemblies of patients and individuals, higher quality than the Human Genome Project, and we're discovering variation by assembly comparison, not by mapping reads to a reference, which is a completely different exercise now. The really good news of this is that we're discovering a whole new set of genetic variants that we've previously missed. And that is, if some of you know me and hear my talks, it's structural variation. Things that are not simple single base pair changes, but deletions and duplications and insertions that are 10, 20, 50, 70 base pairs, 700 kb in size, present in children. So there's a threefold increase in sensitivity in detection of structural variation and near complete characterization, not understanding, but characterization of all forms of germline variation for the first time. And on top of that, if that wasn't good enough, you also get methylation. Methylation granted from the blood, because we're not biopsying brains in autism, we're getting blood from patients. But this is an example of early work in my lab by 2 clinical fellows. This is a child with Barrett-Scott syndrome, not autism, it's a skeletal dysmorphology. It's caused by a CGG repeat expansion in a 5' UTR of a gene called XYLT1. This is how in 2019, the postdocs, they were starting their own labs, were discovering this by running southern blots. Some of you are old enough to remember what these are, but this is a very painful way to discover a CGG repeat expansion. And so in this case, both copies have to be hypermethylated and expanded for a child to have this developmental defect. This is sequencing of the mom and the child with Hi-Fi PacBio, and I don't think you can see the actual letters, but you'll have to trust me. This is just a CGG hyperexpanded allele in both paternal and maternal complements. This is a premutation allele. So phased, expanded, and then on top of that, methylation. So the red here represents hypermethylation, the blue represents hypomethylation. Proband has 2 alleles that are hyperexpanded, and it has hypermethylation, one-stop shopping, phased genome methylation, diagnosis, clinical test. Kid has Barrett-Scott. You could do this prenatally in principle, right? This is why Illumina goes away. The ability to do this routinely on patients, I believe, is going to be here within 5 years. Okay, so what we've been doing, and this is all unpublished data now, is we've been going back to those families that we haven't solved, which is the majority, but we've been really careful about how we're selecting our first families, because this is still expensive technology, like every genomic technology is. And we're sequencing them with all technologies, Hi-Fi, ONT, PacBio, and we're sequencing as many times as we can quads, just that same design that we had before, and we're picking females that are affected with autism as opposed to males, because our data and others have shown that de novo mutations are twice as likely to be discovered in a female with autism than a male. We don't know why that is, and there's some idea that it takes more insult to push a female over the edge into autism. And so we're selecting female families that we haven't solved where the PRS, which is the polygenic risk score, is insignificant, And so even the polygenic risk people that are looking at common variants would say we can't solve this case. Okay, so that's the backdrop. We are selecting female quad/trio families where every genetic test has failed to find the genetic cause. And there's still lots of them. So our goal is to do 70. We've started production and have the first 50 in production. But here are some preliminary results. Oh, I should mention, we're focusing on structural variation because we know that's largely the unascertained. And we're actually discovering events that are exclusive to either the sibling or the proband. And we're analyzing the frequency of smaller structural variants throughout the whole genome and comparing proband and sibling to see if there's any difference. This is the result thus far, not significant, but it's an N of 9. So 9 autism families, girls with autism. I hope you can see the probands here, and we've broken out in terms of whether the deletion and insertion events are exonic, intergenic, or intronic. There's a consistent trend for the girls with autism to have more private, or at least autism-specific structural variation events than their unaffected siblings. And the unaffected siblings include both males and females, so we're focusing just here on the autism. But this difference of 818 versus 729 is still not significant, and we're hoping, though, with essentially larger screens of patients, we'll get this to a point that we'll believe it. So that's teaser number 1 in terms of potential signal in structural variation genome-wide that's going to be rare, but collecting in the probands, in this case, females with autism. Observation number 2, we've looked at differentially methylated regions. Why? Because we have, from the blood, not the brain, we have methylation status for every region of the genome for the CPGs. And so what my postdoc, this is a new postdoc, Young Sue, has been doing is looking for differential methylated regions between proband and sibling within the same family. And she's identifying anywhere from like 1,000 per family to all the way up to 8,000, which seems ridiculous to me, but those are the numbers. And the striking thing, when she started looking at this data, we are finding genes that we've already implicated in autism, GRIN1, as an example, that show evidence of differentially methylated status across the gene body between the proband and the sibling. So we've analyzed not 9, but 5 families so far for differentially methylated regions in the blood. And this is the preliminary result number 2, or teaser number 2. We are finding essentially in 2 of the 5 families that we've tested, we're finding an enrichment of differentially methylated regions in genes associated with synapse function. And so this is the gene ontology analysis for those 2 families, showing neuron-to-neuron synapse as being significantly enriched in DMRs, postsynaptic density, asymmetric synapse, and postsynaptic specialization. I do not know how this could occur by chance, why it would go specifically to synapse, unless this is a readout, an epigenetic readout. We don't have any genetics yet for this. All we do is we have differentially methylated status. So a colleague of mine said the other day, well, said that doesn't tell you, it could be just a readout of the fact that the child has autism. But if that's the case, this could be incredibly valuable, right, for prenatal, postnatal testing, right, if we could see this kind of signal. So again, this is based on very limited numbers, but it comes from actually doing genome-wide comprehensive assessment, both structural variation as well as methylation. All right, so to summarize, I've talked about really 3 things. The beauty of the de novo mutations to actually help us identify the hundreds of new genes that have been implicated in autism and developmental delay. And I guess the important point to take home is that almost all of those genes that have been identified and reported for autism are not associated with high-functioning IQ cases of autism. They are associated with kids that have typically severe intellectual impairment. There is a super signal that's probably bigger genetically in terms of its contribution to autism coming from private inherited mutations. So these are mutations that are coming into the family from mutations that arose a few generations back in the family history, and the signals get stronger if you require there to be 2 or more of those types of events. and they're coalescing on the same pathways. But the fact that they're transmitting and they're enriched in multiple genes or different genes suggests to us multiple hits are necessary. So digenic, trigenic model, right, for autism. And then the last point is the future one, which is we should get ready for a day, it's here already, where we take long-read sequencing of our patients, comprehensively sequence and assemble the genome, telomere to telomere, to discover all forms of variation and methylation present in those genomes. And when I think about this, this is probably the most significant contribution that can be made to a person's medical record. Because if we sequenced your genome shortly after birth and had complete sequence information, it would be the most valuable thing. Everything from cancer diagnosis when you're 55, all the way to your planning for your children down the road, and your susceptibilities. And as we catalog the variation, and as we begin to understand it, which will take probably decades, you can always go back to your complete sequence and say, oh, by the way, you have a 4-fold increase for hypertension on top of this and on top of that. All right, so it's amazing how far the field has come. I mean, I got to see all this in kind of real time, going from arrays to exomes to genomes. And I think the long-read sequencing is the cusp of a new revolution, I think, in our field. And I guess you could look at it as half full, half empty. Well, you solve perhaps 50% of cases, but you still haven't solved 50% of the cases, right? But I think we will be able to chip away at this. The more complete information we get genetically, right, that comes from methylation, that comes from structural variation, and so on. All right, so I'll stop there. I just want to acknowledge these folks, especially the ones I've highlighted. I presented the work largely of 3 postdocs. 2 now are their own bosses. Tin Yun Wang has gone back to China. Amy Wilford actually works for a company outside of Seattle now. And Young Su is a new young postdoc. And also just great collaborators all around the world who have contributed usually with patient samples, but not just patient samples, actually recontact to actually identify the phenotypes associated with those patients. And so those clinical net connections have always been fantastic and a great part of the work we do, in the fact that we can reach the families through the clinicians. All right, and of course, last and the most important are the patients and families. For the parents that have the foresight and the courage, right, to actually help with research by providing the material, right, for this type of research. All right, thank you, guys.
Okay. Thank you very much, Evan. I think that's very exciting to learn about what is really cooking in the new perspective of sequencing. I think there will be questions. One question is already is there. If you can, yes, if you can read, you can read it loud, please.
So you're the top one here, is that it? From Stefania. This is a long question. How all of this could improve the life of a person with autism. In other words, we may someday have biomarkers with very other costs. We should do genetic analysis to all infants for early diagnosis, but how to act on possible modification of genes of DNA to modify a person's typical development. Still, research is being done in the mouse models. It seems to me that we are still too far from being able to give answers to autistic persons and families. What are your thoughts on this? Okay, so there's a couple things. I guess let me paraphrase this and simply say, I guess one question is embedded in this, you can't really solve any of these cases with treatments. So if you find these associations or you find these genes, why is it useful for the family? And yeah, it's a great question. It's the same question parents often ask us when they actually, the case has been solved. And everyone who's a clinician or has clinician friends knows that the right answer is that discovery of the mutation is only the beginning, and that does not mean there's a cure. In fact, we never talk about cures with children with autism. But one thing I would tell you, people often say this is not actionable. That is true, because there's not a drug you can give, and it's very hard to envision how something that's early in development could be treated so early right now, right, to make it a differential outcome. But one thing that is incredibly valuable is having families know why their child has autism. And I can't tell you how many families have said, the fact that I know that it wasn't that third glass of wine I drank when I was pregnant in the first trimester, and it wasn't because I rolled my truck and had a car accident, that it wasn't because I was exposed to this chemical, that it was because of this de novo mutation in a gene, that provides for a lot of families actually some peace of mind, in part of just because they know they can have additional children and not worry about necessary transmission because it's a de novo event, right, in those cases. So there's huge value, psychologically, I would say, for the families to know why their child has autism. There's also something else that is really, I think, many of you probably know, is that once families know they have a specific cause, they find other families that have the same genetic cause. And you can try to prevent it, right, from happening by, not telling them who the clinicians are, because some people want privacy and all these other things, but they will find each other. They form networks and groups, and it's amazing to me to see the things that come out of those groups, those family foundations. It's that kind of like the birth of these family foundations that emerges from that. And what comes out the other end after being with some of these groups for 5 and 10 years is a very sophisticated group of families that not only have support through each other, because they have children that are much more similar than Johnny, who had autism, that is in this support group that is completely different genetic cause. But they also have knowledge. So when there's a drug developed, there's essentially an opportunity to go in for essentially a phase 2. These individuals actually are kind of ahead of the bus, like they're front of the queue, because they have the numbers of patients, maybe 50, 100 families that are now collected, but they're also extremely knowledgeable, and they will reject actually participating in a trial because they know that there's a better drug potentially coming out in 3 months or so on. So I guess my answer is that there is no quick fix and no treatment, and yes, the work on mice is super important. It needs to be done. And I think it'll take decades before there will be treatments, not cures, to improve the quality of life of these children. But this is like, there's huge value to actually identifying the genetic causes, or maybe even the epigenetic causes, to start to group and coalesce families, because it is such a heterogeneous disorder. But yeah, it's a great question, and it's fully loaded question, because there's probably not a right answer, but this is what I have observed, having worked in this field for, I guess now 30 years. Is there anyone else?
Yes. I guess there is a question from Elena Bonora. Elena, can you open your microphone?
Yes, thank you. First, it's always fantastic to listen to your presentation, and really inspiring. And I have a few questions. One is when you showed the private genes, there was the network of BRAF connected with P10, which are also kind of oncogenes or oncology-related genes. I was wondering if there is any report for the people that carried variant in these genes and ASD, they also are at risk of developing some kind of cancer-related syndrome, or?
Yeah, no, it's a good question, and I have to tell you that all I can tell you is we haven't pursued a study this way. Obviously, you need longitudinal data for that type of study. We know anecdotes of that's the case. So I'm thinking particularly on early on when we discovered CHD8. I think some of you know CHD8 is a very, of the genes that have been implicated, it's one of the enriched in autism, not exclusive to autism, but it's enriched in autism. And it's, before we discovered it, it was already implicated, essentially associated with gastrointestinal cancer. And we had, I think, one family where there was an inherited case of it. So we went right away back and we had the DNA. We went back to check the father because we wanted to retype it. We wanted to look to see if it was mosaic, if it was truly germline, all those things. But the father had died of cancer, gastrointestinal cancer. And so that was an N of 1, it was an observation. But it brought up the idea that, yes, I mean, these genes, many of those genes are genes associated with essentially oncogenes, some of them are tumor suppressors. And if a child has that de novo mutation or has inherited that mutation, the child that is inherited in a Knudsen model would already have one hit toward essentially developing it. But yeah, I think it's a study worth doing, but it has to be done very, very carefully. I was also told by a clinician is that these families have other things to worry about right now.
No, of course.
And not to like bring up this possibility. So, yeah. That was one of the first things. It's not just P10 and CHD, there's a whole raft, especially of these chromatin remodelers that are associated with cancer. But yeah, it's a great thing to pursue, maybe with these large biobanks, right? If you can start finding those cases.
Yeah, like comorbidities, etc. would be. And the second question is the methylation analysis, because I know that with the long reads it's fantastic that in single experiment you can see so many things. But we know that also methylation can change across age. So I was wondering, do you think should be resampled the methylation? I mean, you have found very interesting data now on the methylation on the synaptic genes.
Yeah, no, it's a great idea, right? To sample longitudinally to see if it's changing. Is that what you're thinking?
Yes, yeah.
Yeah. No,
I know it's still not practical in terms of cost, but I think.
Yeah, although I have to say, the long-read sequencing technology, the ONT, is particularly sensitive for methylation and also a little bit cheaper than the hi-fi, right? So you could do this if you had the setup. The other question I have is what is going on in all the other tissues, right? I would love to know what is happening in fibroblasts, right? Or get IPS material or derive IPS and actually differentiate neurons, right? Because it's blood in the end that we're looking at, right? So the fact that we're seeing, and again, it's only 2 families out of the 5, right? So there's all very small numbers. But we're seeing it in the blood, and obviously the organ of interest here is the developing brain, right? But yeah, no, it's a great idea.
Okay, thank you.
Yeah,
and
thank you, Ayala.
Thank you again for your wonderful talk.
Nice to see you again.
Hi.
Evan, this morning we were just referring to the most recent paper which came out of after the conference you were moderating in May on the pangenome story. And this the paper on the Y chromosome.
Oh, yes.
Can you comment a little bit on, I mean, what are the advantages now that you have this complete sequence or full chromosome, the Y chromosome?
Well, so to be fair, right, to be careful, the Y chromosome is nearly complete. For those of you who are unfamiliar with what some people think is a worthless chromosome, the Y chromosome has a very large QH region, which is made up of satellites that go on for many megabases. And in that paper, the ones that just came out, that region typically is not fully assembled, right? So there's, just to be sure, most of the Y is assembled, but not that part, right? So it's like, I would say 90% of the Y is complete. But with that caveat, I think it's actually, to me, it's interesting. Like, biologically and from a perspective of population evolution, it's very, very interesting what changes have occurred, because the Y lives its entire life in a male germline, right, and has subjected to a higher rate of mutation. It doesn't have homology in the typical sense other than the power regions. In terms of biomedical impact, it's fairly limited, I think, still, right? And I think what's nice about it, though, is that people can start to do associations, particularly with respect to fertility, right? And I'm sure there will be many new cases of infertility that will be solved. We are going to look at it with respect to these families as well. But remember, we selected females for study for proband. So we're going back and starting to do some males as well. But I think the Y chromosome is interesting from the perspective of trying to be complete. And the fact that we can sequence the Y means that we can sequence pretty much everything, right, in the genome. But yeah.
Thank you. If there are no other questions, I think we can do two things. Just relax a little bit and then go for the last talk of the series, which is Daniel Gershwind recorded already. So is there I think, Domenico, do you think we have some coffee or something? Yes? Oh, yes. We can stop for 10 minutes and then come back for the last talk. Okay? Thank you again. Thank you. Thank you.
Ho caricato ho messo una cartellina dove per giorno, quindi trovi questa cartellina qua che vedi, dobbiamo chiedergli permesso, sì.
Non era proprio, quindi i nomi non si leggevano.
Ok, gli chiediamo.
C'è la registrazione, se la risentono, poi se la legge, però senza leggere i nomi.
Se hai le slide, certo.
Adesso mi marito il tuo numero.
Sì, volentieri.
Perché noi facciamo questo mega progetto sulla FISA, la cascata del Colonial. Quindi se dovesse essere coinvolti sarebbe una cosa.
Io sono molto contento se c'è qualcosa che possiamo fare usando dei modelli magari piuttosto che analisi.
To sit here wherever you want. Yeah. The good news is that in the afternoon and during the talk of Evan Eichler is that the number of people following in remote were about 50, right? 50 persons following in remote, which is good news because this morning we had quite a crisis with problems, with technical problems. Good. Now you introduce the last speaker?
We have a last speaker who is Daniel Geschwinde from the UCLA in USA. Daniel is actually traveling now, is on a plane, and he kindly pre-recorded his talk, so I will just start. Here we are. Okay. Non sono sicuro che riusciamo a sentire. Ah, ecco, ok, giusto.
It's really a great pleasure to be able to speak at this first international forum on ASD in Calabria, Italy.
I just switch off the microphone. Our microphone. Ok. Non sentono?
I'm very sorry I can't be there in person. You have no idea how interesting it would be to travel and be with you, but unfortunately I can't. But I'll talk with you today about my point of view on autism genetics with this title, On the Frontiers of Precision Medicine. And of course, autism has been a kind of neuropsychiatric condition since 1947. But I'm going to talk about a different kind of analysis today, not the usual Freudian analysis, but we're talking about DNA analysis and hopefully not so much on the couch. What I'm going to talk about today, first I'm going to introduce the notion of precision health. We're in the midst of a genetics revolution in medicine, and I will introduce that. Next, I will discuss what genes have been identified in autism, and then how we move from individual genes to biological understanding and treatment. And my disclosures are below. I'm not talking about anything related to those industry relationships today. So the goal of precision medicine is to deliver the right treatment every time to the right person. But of course, prediction is very difficult, especially if it's about the future. So how do we do this? So we're not saying this is easy, but it's a whole new idea. It's a way of looking at medicine that's fundamentally different than the way we do it now. And the major goals of precision medicine are to use genetics and genomics and big data to diagnose, to predict both disease risk so we can prevent, as well as understand the best treatment to get the best outcome. Prevention is a critical part of precision medicine. You can imagine if when everybody's born, we have their whole genome sequence, there's a lot that we can predict. Now we have a long way to go, but this is the promise of precision medicine. Now another thing, those of us who study genetics, one of our main motivations is to understand disease pathophysiology. In other words, genetics gives you a causal starting point or anchor from which to understand disease mechanisms and develop treatments. So, but this has always been a critical part of medicine. So this new revolution in healthcare, we have medicine today. It's reactive, it's population-based. It's kind of one-size-fits-all model of care. Precision health is predictive, preventive, patient-centric model of care. You wouldn't just wear any pair of glasses. You don't just take a pair of glasses off the shelf. Your prescription is tailored to your vision. But there are very few areas of medicine where that's actually done. It's a paradigm that emphasizes the ways in which disease risks are unique and different, just like other more obvious characteristics. Now, this has been really, kind of amazing in areas like cancer, where, so we know that most drugs prescribed in the United States are not that effective. Now, in cancer, precision medicine has really changed therapy in that we now sequence the tumors to identify somatic mutations, that is, mutations in the tumor that may be driving the cancers and that will respond to very specific medicines. But the same is happening in other areas as well. And there are two major drivers of this. Dramatically decreasing DNA sequencing costs alter the approach to human disease as computer power increases. So what I'm showing on the left is Moore's law. Gordon Moore predicted in I think it was the 60s, that every 18 months there would be a doubling in the power of an integrated circuit due to the ability to pack more transistors onto an integrated chip. And he said that would go for 10 years. Well, in that case, he was wrong. There's basically been an annual doubling since he made that statement. This is what has led to the iPhone and everything else, holding computers in our hands. You can see that is an exponential decline. This is a log scale. Something happened here in the mid-2000s where actually sequencing costs dropped even more exponentially. And this is next generation sequencing. So now a whole genome sequence costs less than $1,000, and there'll be a time when a whole genome sequence will cost the price of a typical set of lab tests that you get, under 100 euros. Millions of genomes are sequenced, and we now have the power to analyze them. So we're going to be we're at the beginning, really, of what's going to be an explosion of genetic discoveries across populations. The cost of genome sequencing continues to drop rapidly. More human genomes being sequenced, a more accurate understanding of disease. Now, as Evan Eichler is likely going to tell you, I think he's talking there, we're not sequencing the whole human genome. There are kind of technological advances that are necessary. But we can sequence enough that it becomes diagnostically very, very powerful. So most of the time in cancer right now, targeted sequencing is done. Various sequencing that identify somatic mosaicism within cancer, because not every cancer cell in a tissue will have the mutation, so you need to sequence very deep. But this might sequence 500 genes. Whole exome sequencing, where you sequence the protein coding part of the genome, it's about 4%, 2 to 4% of the genome is coding for proteins. And we can interpret that very well. Not perfectly, but there's a lot that we can learn from that. So that's been the focus of most clinical sequencing, just by sequencing 2% to 3% of the genome. We're moving towards a time very soon where we're going to stop doing all of these and just do whole genome sequencing because the technology is getting so cheap. And here you cover the whole genome. The difficulty is that we don't know what a lot of the non-coding parts of the genome do, so we have a lot to learn. But this is going to be the standard soon. But most of what we know is from whole exome sequencing. And this is from a few years ago from UCLA. At UCLA, my colleague Stan Nelson was one of the first in the country, second to Baylor, to start doing whole exome sequencing to diagnose people with undiagnosed diseases. And this is from my colleague Brent Fogel, who's a neurogeneticist, where we just have the rates of neurologic diagnosis in a number of different papers. You can see that the rate is very, very high if the patients are chosen with certain kinds of ataxias or dementias or neurodegenerative syndromes, etc. It's even higher in children with cardiac or developmental abnormalities. It's a very high yield clinical test, and these rare Mendelian forms of disease are estimated to account for 7% of patients overall. So even though the disorders are individually rare, when they're grouped together, it's a substantial population. And when you talk about developmental disorders writ large, broadly, it may be, we're going to be able to identify, 20%, 30% using this technology. And the implications of knowing a genetic cause, even today, where we're at the beginning of this, are quite significant. For example, is this a new mutation, like Down syndrome, that occurs in the germline, in the egg, in the gamete, sperm or the egg, or is it a germline mutation? If it's germline, it's more likely to be heritable. If it's a new mutation, it's far less likely to be heritable. That has enormous implications for recurrence risk. How is it inherited? Is it recessive? Is it dominant? Etc. All of that can teach one a lot and be very important to families. But knowing the genetic basis of the disorder has potentially significant implications for treatment and prevention as well. You can identify the mechanism of disease and target therapy, like in cancer. 6 to 7% of cancers now have a therapy that's directly targeted based on the genetic mutation. The mechanism of mutation may be preventable. By doing this sequencing early, we save patients and the medical system actually thousands of dollars and a lot of time and hassle. So, this is what's happening in terms of the revolution in medicine. How does this apply to autism and neuropsychiatric disorders? Here, Jonathan Flint and I list the prevalence, lifetime prevalence of psychiatric disorders from anxiety, major depression, down to autism, third from the bottom. More than 1%, not a rare disorder. These are common disorders. They have high...
I just need to share again.
Hello, everybody. I'm really... The GRECA word is this. And something is called... Most of the population, ancestral populations, this is from...
Si è scollegato proprio.
Adesso sì. No,
vai indietro.
We're able to identify. And this is really moving the field forward. So as we get large cohorts and we're sequencing large populations, we're identifying large numbers of genetic risk. But what does this mean from a theoretical standpoint? Let me back up a second. Humans have grown quickly from very small ancestral populations. This is from my favorite scientific source, Wikipedia. But basically, most of the population was very, very small until about 2,500 years ago, and then there was a sudden dramatic increase. So we come from a very small population, a kind of bottleneck, a series of bottlenecks in a way, and now so what this means is that when we look at genetic variation, most of the stuff that's common is shared across human populations, quite a bit of it. This is just showing the history of humans in a cartoon. This is from Jim Lovsky about a decade ago. It's a beautiful diagram. The ancient things come from our shared ancestors a long time ago. But they have small effects with respect to disease because natural selection, evolution has worked to remove strong bad actors. And in brain genes, even more so, they're under strong purifying selection. So that when we look for things where an individual mutation can have a big effect, it's something that's much more likely to be new or very recent. These things are going to have larger effect because evolution and natural selection hasn't had time to act on them quite as much. And so common variants are going to have small effects. Rare variants might have small effects, but they have the possibility of having large effects. And so to put that into a graph visually, there's a kind of, I don't want to use the word trade-off, but you can see effect size on one axis and frequency of the mutation or the genetic variation in populations. And something is called a mutation when it's rare, less than 1%, and it's generally sufficient to be causal. Because something's rare does not make it a mutation, and it has to be studied. But mutations are generally rare. They have large effect and can cause disease. And common variants that are greater than 1% they have very small effects. They've been weeded out by natural selection. So they contribute to disease in an aggregate fashion. One is not enough. You might have to have 100 or 2,000 to have very high risk for a disease. And I'll show that later. So we can genotype common genetic variation for about $30 or $40 using a microarray. But to get to rare mutations, we need to do sequencing. And so how does this work? How can common variations add up? And it's basically thought of in this way. Something called polygenicity. That is, you need many, many, many genes of small effect size. Baseline risk would be in the middle. You're at decreased risk if you have very little risk. And if you carry many of these small effect size, you're going to have increased risk. So you could identify the 5 or 1% of the population at highest risk with this kind of polygenic additive risk. And maybe in here, all the way on the right, are the people with the very, very large effect size mutations, where only one is needed. So when we're beginning to do studies of autism, we have to take this into account. Are we looking to find rare, new, de novo mutations like Down syndrome? The parents don't have it. It's genetic, but it's not inherited. It's a mutation that occurs in the egg or the sperm. Or are we looking for inherited factors? And so different studies will be powered to find different things. So in the Simon's collections, called the Simon Simplex collection, or SPARC, in the Simplex collection, it's all families like this, where there's no other history of autism, and it's actually been screened out. So it's like a lightning bolt. It's a rare event. And you collect the father, the mother, the proband, and a sibling. In the SPARC, about 10 to 20% are multiplex, where you have 2 or more children with autism. The parents don't necessarily have it because having autism reduces fecundity, but they may have some minor kind of what we call the broader phenotype. Well, and so the GRECA cohort is this. It's something that we started about 20 years ago. So AGREE was founded in 1998, an open resource, more than 1,500 families and 6,000 individuals, large-scale genetic and phenotype data. And this was the first genetic resource, and it was focused on finding inherited variation by collecting multiplex families, families with 2 or more children with autism, which is probably only about 10 or 15%, maybe 20% of autism. And then there was the Simon Simplex collection and SPARC, and you're going to probably hear more about those today. I'm not going to talk about them a lot. But this was focused on families that just had one affected and no other history of autism. And both of these have made big contributions. I would say the Simons, in some ways, has been a bigger contribution to date. So, because most of the rapid growth in genes comes from identifying genes with large effect, where a single mutation is probably sufficient to have autism. And this is due to the whole exome sequencing. And again, the point I'm making here that Brett Abrams and I made about 15 years ago is that genetics is very technology driven, our ability to identify things. And as the technology has exploded and become cheap, what's actually very expensive is phenotyping and collecting patients. It's almost becoming cheaper to do the genetic analysis than it is to collect patients for studies. But all of this work has led to a vast increase in our understanding of what causes autism. And I'll discuss that on the next slide. So in 2008, a handful of genes, little understanding of mechanism, major pharmaceutical companies withdrawing, little interest. By 2016, more than 200 candidate genes, now more than 200 high confidence risk genes, much clearer mechanistic models, many advances, major drug development efforts going on. So there's a hope for a new neurobiology of autism now due to genetics. And kind of what's been identified are, again, based on technologies that have been applied. So initially, microarrays were used to screen for copy number variations, and these copy number variants in genes were identified here. These are named by sometimes the gene that's deleted or the region that's deleted, etc. We have things like velocardiofacial syndrome, which is a 22q deletion, or DeGeorge syndrome, which is what it's called. And again, that's a very common medical genetic condition that underlies maybe a small percentage of autism. But the sequencing results led to identification of a number, now dozens and dozens of rare genes. But the point here I want to make is none of these are more than 1%. So this component of autism that's due to rare mutations is a collection of rare disorders. And what was known at the time in 2016, it's not too much more now, was that this accounted for maybe 5 or 6% of autism. Together, you'd have the de novo, 5 or 6%, rare inherited variation, a small percent, non-additive effects, etc., recessive, and predicted common inherited is 50%, and then there's a large amount unaccounted for. And that still remains a mystery, although we think that some of that could just be rare genetic variation that will be picked up with whole genome sequencing. So we're identifying many genes. It's predicted there could be certainly more than 500 and maybe more than 1,000. There are highly additive effects when it comes to the predicted common inherited portion. None of the rare variants account for more than 1% of cases. And there's strong pleiotropy. And what pleiotropy means is that you can see that, especially for these medical genetic syndromes, they don't only increase the risk for autism, they increase the risk for many neurodevelopmental outcomes, such as epilepsy or intellectual disability, etc. And we'll talk about that a little bit later. But genetic testing can currently identify about 20% of mutations contributed, in 20% of patients, let's say. So what we do clinically is we do a clinical microarray. If it's a boy, we do Fragile X testing. And then we'll do whole exome, followed by whole genome sequencing. And we think we have about a 15 to 20% yield when we do this, when somebody has autism. So it's clinically indicated, and we do this at UCLA. And again, with most of these large effect variants here and these medical genetic syndromes at the bottom, we see that rare variants don't just cause autism, they lead to a number of neurodevelopmental comorbidities. So what you see here is from a very nice paper published now 6 years ago, where you can see as the number of comorbidities increases, the rate of having a rare de novo event shown on the y-axis increases dramatically. So in other words, if you have delayed motor function, seizures, and intellectual disability, the rate at which we're going to much more likely find a rare genetic variation in you, a rare de novo event, 5 times more than somebody who doesn't have any of those. But still, even in somebody who doesn't have any of those, the odds ratio is still very substantial. And these are just some more other papers that have kind of shown this kind of pleiotropy. For the genetic architecture of autism, again, we don't know much about the rare inherited or predicted common inherited yet, which I'll get to. But we're beginning to know a lot about the genetic contributions on the left, rare de novo mutations. So autism is highly heritable, and I haven't told you much about that yet. But de novo variants is what we've learned most about, and they're isolated in simplex families, and they've driven risk gene discovery. And again, at this point, more than 200 high-confidence genes identified. There are 5 genome-wide significant GWAS loci identified. And there's a modest contribution of rare variants to phenotypic variation. about 6%. That's what's being. So this is a paper from Cell that was published last year. It has its issues, but I think in general it's showing that ASD-associated rare variants are identified in maybe up to 15% of patients. I think they took some license here with what they call a causal rare variant, but nevertheless, I think this is likely to be correct anyway. So most of what we know about autism really comes from this rare variation. And it's relatively easy to understand how disease risk can come from one of those large effect mutations like Down syndrome, 15q duplication, the Nerexin, DeGeorge syndrome, and CHDA, ADNP, all those other mutations I mentioned to you. But how about the additive effects of many common genetic variants, each a small effect? How can they do that? And so again, we go back to our cartoon that you increase risk and that you can identify those with very high risk. Well, what does that mean if you're in the top 5 or 1% of population at highest polygenic additive risk for a condition where you don't have a major mutation, but a combination of common variation and probably some rare variation leads to this increase? Does that give you an odds ratio of 5 or 3? Well, there's been some work in cardiovascular disease showing that this can be quite meaningful. So here's genome-wide polygenic score. That's the additive common variant effect for coronary artery disease. And this is showing that those in the top 0.5% at that level have a 5-fold increase. Those at 2.5%, 4-fold. And those at 8%, that's not even, almost in the top 10%, have a 3-fold increase. So it's really, really substantial. You can see the risk and percentage going up exponentially there. So that these people in this top 5 or 10% of risk of cardiovascular disease, even though they don't have a major mutation, the effect is as if they have a major mutation because of the burden of polygenic risk. And this is just showing some of those numbers here from that paper. So how about common inherited variation in autism? This is the most recent GWAS. There's a larger one going on now. This is with about 25,000 patients, published in Nature Genetics from the Psychiatric GWAS Consortium. The Psychiatric Genetics Consortium, but it's really a GWAS consortium. And there are only 5 loci that reach genome-wide significance. And actually now, even with a much larger data set, there are only 12. These are showing the 5 loci and the genes that are near them. And, this is definitely an advance, but it's a little underwhelming compared to when we had 25,000 in schizophrenia or 35,000, we had over 100 loci. But even though we only had, let's just summarize for a second. So sequencing in families, especially simplex families, those with only one autistic child, has led to identification of hundreds of risk genes, whereas the polygenic kind of genotyping approach to find common genetic variation has, even though autism has a heritability of 70-80%, hasn't led to nearly as much discovery. However, one can use and look at the additive effects. So even though we have few genome-wide significant loci, we can look at the polygenic score, the additive effect. And so if we look, we can add it up and show that we can separate probands with autism from siblings quite substantially. Then, if we look at people who are carrying rare mutations, they also have a slight, barely significant increase. So again, you can have polygenic risk, you can have a rare mutation, and have a little bit of polygenic risk. And then those who don't carry rare mutations, again, they're the main drivers of the polygenic risk. So you can see that there's a kind of continuum, so to speak, of risk. You can have all polygenic risk. You could have polygenic risk with a little bit of rare variation, or even with a de novo variant, you can still be loaded with polygenic risk. And so autism risk follows a liability threshold model, which is a classic behavioral genetics model. It's that bell curve that I showed you earlier. And I'm just going to, in 2007, in a review paper, I kind of illustrated this maybe in a more specific way for autism. And then at that time, autism had three major components: the language component, the social behavior component, and the repetitive restrictive behavior component. And think about each of those as a normal distribution in the population in terms of behavior. And if you're all the way over in, let's say, the top 2%, you'll have autism. And a number of things can move you over there. Copy number variant mutations, rare exome mutations, or recessive or dominant, as well as a polygenic mix of SNPs that could tip you and move over. One of the issues, though, is that common genetic, the CNVs or large effect mutations move you over on all of these kind of at once for the most part. Whereas polygenic risk is going to work additively and may act differently. In other words, the polygenic risk that loads on language some may be related to social behavior, other may be different, etc. And so what's being shown here is, again, more evidence for this liability threshold model from Autistic et al. In Nature Genetics and from our recent paper. And you can see if you have a damaging de novo mutation, shown here, your polygenic risk score is lower. This is not significant. It's only significant in the males here in this data set. But again, you can see this difference where you're going to be much more likely to be loaded with polygenic score if you're not carrying a damaging mutation. We showed the same thing here, affecteds, and then the unaffected shown here. And so we end up with a kind of model where you can have a rare Mendelian mutation, a rare mutation plus a little bit of genetic, maybe environmental and epigenetic piece of the puzzle. And then in most patients, it's going to be a combination of genetic variation, rare and common, maybe with some environment, although again, that's a very small component. It's a really true additive model. But given this complexity, heterogeneity, the pleiotropy, and heterogeneity meaning there are many, many different forms or genetic forms of autism, and even people with polygenic risk may have different combinations of polygenic risk. Pleiotropy, that is single gene that can cause autism, can also cause intellectual disability with seizures with no autism. So how do we begin to connect genotype to phenotype? And so this is again from Autistic et al., very interesting paper in Nature Genetics, where they basically show that different mutation kind of patterns impact phenotypes differently. Again, some of this is just confirming other earlier studies, but it's very, very important to kind of continue to look at this as our samples get bigger and bigger. And so we have the social domains up here, and those are loading primarily on the polygenic score for autism, not as much for schizophrenia. So again, the social component very strongly correlated with the polygenic risk score. And these de novo large effect mutations, whether they're missense or loss of function, are much more likely to impact motor and adaptive function. So we recently did a study of agree multiplex families. We've been working with them for a long time. And recently this paper was published in PNAS, led by Matilda Cirnigliaro, who is actually from Sicily, from Catania. And she's been in my lab now for 4 years and has done beautiful work with Tim Cheng and Stephanie Ortega. So now we took 1,000 multiplex families from Agree, did genome sequencing, and ended up looking at the rate of rare de novo and rare inherited. And previously, in another paper in Cell, Elizabeth Russo and Laura Perez-Cano had shown that when you look at multiplex families, the actual contribution of rare de novo variants is much lower than in the simplex families. But we reasoned that by enriching for families that more likely to have inherited autism because they have multiple affecteds versus simplex, where it's more likely to be de novo, we might have increased power to find rare inherited variation. And that's shown here. In this small study, remember only about under 2,000 probands, roughly, we identified 7 novel risk genes that are mostly supported by rare inherited variation. So if we look here to the right, this is just a statistical score. Above the bar is significant. These are the new genes that are candidate risk genes. Again, they need to be replicated in other studies. But it's interesting if you look at the proportion of inherited protein truncating variants, the vast majority of these, this one very, very low. But in 6 out of 7, quite significant contribution from inherited rare protein truncating variants. And again, that's shown here. You can see a lot of green. So this study, we had 2,100 autism cases, 1,000 multiplex families, 7 risk genes. In the latest SPARK study, which a number of us were involved in, and I know Evan Eichler played a major role in this, there are 43,000 autism cases, over 29,000 families, about 20% multiplex, but only 5 new risk genes. And most of the new risk genes were supported by inherited rare variation. So one of the points I want to make is that how you collect your families is going to determine what you're going to find. And if we don't collect families and study autism that's inherited, we're not going to find inherited variation. But it's hard to find, and we need much larger sample sizes. So getting back to precision health, I think large-scale, countrywide, or health system-wide analysis of autism is going to be necessary, so that no longer will we be doing small studies where we collect patients. We'll just phenotyping or sequencing everybody in the medical system and pulling out those with autism and being able to study them. And there, I think, we're going to find a lot of what we're missing. So in this study, there were 74 risk genes at an FDR less than 0.1. Again, that's not the most stringent statistical criteria, but we use that to kind of identify as many candidate genes as possible, and then we'll rely on replication in future studies. So about half of them have no inherited component, but about half of them have an inherited component. And in fact, in about a quarter, the inherited component comprises more than 50% of the risk. So again, this importance of kind of recognizing that you're going to find what you're looking for in some ways, depending on your study design. But I think the most interesting part of this paper was some beautiful work that Matilda did, looking at how in these families that have inherited rare variation, as well as polygenic risk, how the two come together. And I showed you briefly before that there was a separation. But how are we able to use these families and to leverage their structure to understand autism better, even with this complex additive genetic risk? What we did is we began to ask, if we just look at autism polygenic risk, we compared it to schizophrenia, educational attainment, we don't see any of this with those. We can see that if we look at language delay, as defined by age of first word in months, or age at first phrase in months, so it would be a 2-word phrase. And we can look at those who are carrying who are affected with who have autism, and we're looking at the inheritance of common variation, we can see that it's being inherited to those both of these have autism here, right? Both of these have autism. They're not carrying a rare de novo variant, but they are carrying polygenic risk, but only if they're in the group of significant language delay, shown here. So what this tells us is that polygenic risk for autism is also somehow increasing risk for language delay. And this is interesting to me because language development in DSM-5 was taken out of the diagnostic criteria for autism. And this genetics first kind of approach would tell us that no, language is fundamental. And this language delay, or whatever it represents, is a fundamental core aspect of autism. So, again, autism in the individual. You can have a Mendelian model, rare. You can have oligogenic, two-hit rare variant. You can have even a combination of these. And in fact, when we think about why the parents are unaffected, even though they are giving a rare inherited variant to their child, as we showed in the study I just showed you, and the parents don't have autism, it's because there's sub-threshold that the polygenic risk has been transmitted to the child in addition to the rare inherited variant. And again, it's an additive model. And so you need to get over a certain threshold to have substantial risk for autism. But the challenge here again is that advances in genetics and genomics have identified many genes involved in susceptibility for autism. These genes provide targets for understanding the disease mechanisms and direct therapeutic development. However, they highlight extreme heterogeneity. Are we going to have to develop a specific treatment for each disorder? Or will there be convergence in specific biological pathways, developmental stages, or brain circuitry? And let me give you an analogy here. Cancer. Cancer is a disorder of proliferation of cells and their invasion into other tissues. All cancers do that. That's their pathophysiology. And in fact, the first therapies for cancer came by understanding the proliferation and using antimitotic agents as well as radiation to kill rapidly dividing, proliferating cells. Now, of course, that led to side effects, but it was better than no treatment at all. Now, cancer is moving back to individual genes and pathways. So it's a really interesting question. I would love to understand autism at the high level of proliferation and invasion. But in the end, maybe we're really going to need a precision medicine approach where we identify mutations, and those mutations tell us what the therapies are. And different classes of mutations have different therapies. So again, this is, again, from almost 15 years ago, but I think it still stays today. There were some known, on the left, are some known causes of autism here. These were mostly medical genetic syndromes or copy number variation, duplication 15q being one of the most penetrant. Almost 85% of the kids with this have autism. So this is the molecular level. Genetic variants to molecular pathways. So maybe there's some overlap of molecular pathways here that can be targeted just like in cancer. Or maybe it's at the level of biological pathways. Abnormal neuronal migration, patterning, faulty wiring, etc. We know that at some level autism has to involve circuits that mediate social cognition in humans. But that's a pretty wide set of circuits. It's going to involve a large part of the brain. So it's not going to be a very specific circuit disorder, probably. because you can disrupt social cognition so many ways. The main part of the social brain is in the frontal and anterior temporal lobes, but they're connected to so many other areas, and those connections are so critical that it really likely involves a large swath of brain. And most of these mutations, the genes involved, are expressed throughout the brain. So one way to get autism is to just affect the connections between different areas of the brain in a subtle way, so that there's massive changes in timing and difficulty in doing things that require rapid, kind of in-the-moment learning and implicit learning, like language and social cognition. But we and other people started to ask, as autism genes were being identified, when and where do they act? Is there some convergence somewhere? So I'm showing brain development here, weeks, birth is right there. This is mid-gestation. These are the biological processes that are maximum at those times. And one can see that the neurons are born in the brain until about 24 weeks. They migrate till about birth. There's axon outgrowth that continues post-birth, and synapses are produced throughout development and are actually eliminated after 2 years of age. So autism genes, it turns out, these de novo large effect mutations impact genes that affect gene regulation, transcription factors, and are expressed during early embryonic development. So the transcriptional regulators are expressed early here. Genes involved in synaptic development that are starting here are also expressed fetally. And when we look at which layers they impact, it's primarily the superficial layers. There are several genes that impact layer 5 and 6 neurons, don't get me wrong, but it's this enrichment here. And what's interesting about that is that those are the regions that connect the two hemispheres with each other and are responsible for long-range connectivity in the cerebral cortex. So if things are going this early, how do we develop therapeutics? It's a really tough issue. One possibility, and I believe this is going to occur in the next 10 or 15 years, is in utero gene therapy, targeting specific genes. But in the meantime, we have a lot of mechanism to understand. So how do we develop therapeutics? One of the big advances has been the ability to take skin or blood from any patient with any disease or control to reprogram into a pluripotent cell. What that means is that cell can become any tissue in the body. And you can make little brains and little neurons in a tissue culture dish from a patient. And the first time this was used was in autism, was in Ricardo Dolmetsch's lab and work that was led by Sergio Pasco, using neurons from patients to identify the physiologic deficits associated with Timothy syndrome, and then to use that for treatment development. So we think this has large promise, that if you look at this kind of trade-off between mechanistic insight, human relevance, and throughput, which means the number of genes you can actually study. You can't study many genes in mouse models or in primates or in live humans. They're not that human relevant, as you can see. There's this trade-off. iPSCs allow for studying many genes in parallel. That's high throughput. Good mechanistic insight because you can manipulate the system, and high human relevance. So this is one of the more recent studies led by now Sergio Pasco in his own laboratory at Stanford, where years and years of study in dozens of lines of patients with 22q deletion syndrome. So a total of 43 lines, an enormous study. And we can grow these in a dish and make three-dimensional organoids. And from these, after a day 100 or so, we do transcriptional analysis calcium imaging and patch clamping to look at physiology. And here what I'm showing is that we did gene expression analysis, which predicts deficit in voltage-gated calcium channel activity, both at 75 and 100 days, and early on, serious deficits in calcium-mediated signaling. So we can go, and Sergio's lab with John Huguenard went into the dish, and they can see both with 22q deletion and with one of the major genes in the region, which is a microRNA, you end up leading to this decreased excitability with the application of potassium to the bath. Here we can rescue that with a drug called raclopride, shown here in the dish. So this is a proof of principle. It does not, we're not going to use this drug necessarily in patients, but it tells us that we can identify the consequences of these mutations. We can study it, understand the mechanism, and treat it. So now Sergio's lab is using this to study and develop drugs for these mutations. We made a mouse model of one of the early genes identified, which is a recessive mutation in the Amish called CNGNAP2. And we had done previous work to show that this causes behavioral deficits in the mouse, like autism. And Olga Penagarin-Cano had shown that when we give oxytocin, it can acutely normalize social behavior in a mouse. It lasts about an hour. So we studied the brain mechanisms. Now that we know that we have a mouse that has abnormal social behavior and repetitive behavior, and we can reverse that with a drug, we can study that using fMRI and activity mapping in cells. And we can show in the mouse, using fMRI, altered functional connectivity of brain regions, very similar to what happens in patients. We give oxytocin, or we stimulate the hypothalamic oxytocin neurons, and we show normalization of functional connectivity, especially functional connectivity of regions known to be involved in social behavior. We identified a hub brain region called the nucleus accumbens that looked like it was driving this, and we're able to pharmacologically rescue the behavior using a compound TGOT, as well as by stimulating using optogenetics in that region, the nucleus accumbens, to identify, to normalize behavior, as shown here. So, again, we can use animal models, but again, we can really only study one gene at a time in them. The cell models are better for studying many genes. And how do we bridge this? And then the question is, does oxytocin improve autism? Well, the problem is, we had shown that there was an oxytocin deficit in this mouse model. Not everybody with autism is going to have the same problem. So we do not expect that treating everybody with autism with oxytocin is going to help. But there was a big trial done in New England Journal of Medicine last year, 2 years ago now. Children 3 to 17 years of age, 290 people, so a very large study. Intranasal oxytocin twice daily. Outcome, single social withdrawal scale on this thing called the ABC. No improvement with oxytocin. So I wrote an editorial, down but not out. A wide mix of ages. I mean, these kids, it's, from literally children to adulthood. The dosing is not sufficient. Oxytocin has a half-life, functional half-life, of about an hour. So you're giving it twice daily. So it's not really going to do much. So outcome measures, I could go in over and over. But a major issue is the need for coupling the treatment with behavioral therapy. So one of the key issues is that, you're just giving a treatment without any behavior. And the analogy is as follows. Let's say I want to teach somebody to read, but the lights are off in the room. I need to turn the lights on. That's giving the drug. But I turn the lights on. I can't just throw the book on the table. I have to sit with the child or person and teach them how to read. So turning on the light plus behavioral therapy is necessary. And I believe that autism trials have to become a lot more sophisticated. And of course, lastly, not the correct group of patients. We don't know that these people have any oxytocin deficit whatsoever. So a critical issue, I think, is patient selection, proper outcome measures and biomarkers, and the need, in many cases, for coupling the treatment with a behavioral therapy. I'm not saying this in a negative way, I'm saying it in a positive way. We have to learn from our early mistakes, and going forward in therapeutics is going to be hard, and the trials are going to have to be carefully designed. But the genetics and the genetic findings are leading to discovery of biology that's tractable and treatable. So here I'm just showing a few from a review that we wrote several years ago. Excitation inhibition down here with some of the synaptic molecules, the mTOR pathway, which is used, which is targetable, functional connectivity, maybe less targetable. But there are a lot of molecular processes that are targetable. The protein degradation and homeostasis, protein translation, neuronal activity at various channels that are known to be mutated, as well as maybe even neuronal cell adhesion. And again, this latter may have to be a developmental treatment. We just don't know. But one point is that genes don't act alone. They act together in networks and pathways. So downstream of these major gene mutations are hundreds of genes that are disrupted. And actually, we've identified patterns of genes that are disrupted in autism post-mortem brain. So a big question is, can we target those patterns or networks? Can we do that, or do we have to work on individual genes? The answer is, I think we can. So you can take a molecular profile, one of those networks, and you can match that to a drug. And the way you do that is, there's now something called CLUE, or Connectivity Map. It's a database where 1,309 FDA-approved compounds have been given to cells, and gene expression is measured here on the right. Initially using a microarray, now using beads. You can take the gene expression in the disease model from the tissue or patient brain and measure those expression profiles using these technologies, match them to try to reverse, to find a drug that would reverse this pattern, predicted, and then test the drugs in the model systems. One of my students found this recently and showed it to me. I had forgotten I had written this. The recent demonstrations in animal models that certain forms of neurodevelopmental disorders associated with autism can be largely reversed in adulthood represent a paradigm shift in our concept of developmental disorders. Should these findings generalize to humans, genetically identified pathway therapeutics would become the most important area of future treatment research in autism. And again, this means that we may be able to treat people after they're born targeting these pathways and have some success. We just really don't know yet. But in animal models, this has been successful. But I even believe that we're going to be moving into in utero therapy using gene therapy within the next decade. And that's also very exciting. So now we need to translate these data to the individual. The genetic data I showed you is mostly at the population data level in terms of risk. We need to know what comprises disease risk in the individual. And I would say in the last 3 or 4 years, there's been some nice movement in this area by identifying rare inherited variation, identifying de novo mutations, and seeing that there might be a mix, that in an individual you might have to have a combination of mutations. But once that's better defined, we can ask, what, if any, mix of pathways is critical? Are there distinct subgroups of disease that can be grouped together by their genetics? And these questions can be answered by modern genetic technologies. And to understand the effects of a mutation in an individual, we also need very large populations and rigorous statistical analysis. But I'm optimistic, and the field is moving rapidly in this area. So in conclusion, autism risk has a large genetic component, but its etiology is multifactorial and heterogeneous. It's one of the reasons why I called it the autisms about a decade ago, because of this heterogeneity. No two children are alike. and no two children with autism are really alike. But even if we categorize people by their mutations, because people with syndromes do have things in common, of course, and so can we identify different forms of the autisms? Genetic studies have identified more than 100 risk genes, for being very conservative, probably over 200, slightly less conservative. Most are dominant, acting by de novo mechanisms. But I think the study of inherited variation is essential to understand autism. I showed you the example of language delay. But we can capitalize on these findings using convergent evidence from studies in human brain and model systems to begin to elucidate the neural systems basis of autism. So I think, hopefully, I've told you that genetic findings provide a causal starting point to create in vitro and in vivo models, which will allow us to identify potential convergent molecular pathways for development of targeted therapies. So I have a lot of people to thank who worked on this. In the blue are the people in my lab. I mentioned Matilda, Tim, and Stephanie, who did the recent work and agree. Elizabeth, Laura, and Jenny Lowe led the older work. Jenny has been involved in managing these projects for more than a decade in the lab. We have collaborators at Stanford, Dennis Wall on the genetics end, and Sergio Pasca, who's an expert in iPSC and the modeling in a dish. Thomas Waria and Andrew Shurk at the iPsych project in Denmark, which is a population health, mental health initiative. A lot of the stuff that I didn't talk about that underlies our work, identifying brain, the actual pathways in brain involve the other folks on the left, most of whom now run their own laboratories, their own groups, and those people are all starred. And my long-term collaborator, Steve Horvath and Giovanni Coppola, both of whom left UCLA and are now in industry. Our funders, I have to thank NIH, Paul Allen Foundation, Safari, as well as the Lundbeck Foundation. Thank you very much for your attention. I'm very sorry I couldn't be here with you again. I hope the rest of the conference is as fantastic as it looks on paper. Thank you again.
The applause, of course, was also for Professor Bush, has been a hero of the full day, just directing everything. Grazie, Alfredo.
You're welcome. I think we can wrap up and thank the speakers, and we can thank the speakers, I think, and all the people who are attending online, and we can close here. And we see each other tomorrow morning. It's a pity that we are alone here at the dinner.
At dinner, right. 8 p.m., the usual place, the Via Sandro Pertini. That's easy. It's close by. Well, yeah, we'll go. Who wants to go? Whoever wants to go to the hotel, we have transportation by car. Coming back downhill is not a problem. It's just a problem not to run too much because it's easy. And then we'll get together at 8 o'clock in the Via Sandro Pertini, and we can walk together at 8 p.m. Va bene? Grazie,
okay.
Buonasera.
See you.
Grazie. A big applause for our savior of this morning. This morning we had problem with the recording and so on. Grazie.
It's there's postdocs, right?